An octopus has 500 million brain cells, but most of them are not inside its head. Each arm thinks for itself. Each arm tastes the world on its own. It can rewrite its own genetic code in hours, something our bodies cannot do in a lifetime. It matches colors perfectly, yet it is completely colorblind. For 500 million years, this creature has been building a kind of intelligence we are only now beginning to understand. And nearly everything we assumed about it was wrong. If you enjoy this journey, hit like and subscribe so you do not miss what comes next. Now, let us begin. Somewhere in the Pacific Ocean right now, a creature with 3 hearts is hunting in total silence. 2 of those hearts pump blood to its gills. The third pushes blood through the rest of its body. And here is the strange part. When it swims, that third heart stops beating completely. It literally flatlines every time it moves. That is a confirmed scientific fact. This animal prefers to crawl along the ocean floor rather than swim, because swimming is so exhausting that its own heart gives out during the effort. Now, think about what kind of body requires 3 separate hearts just to function. An octopus has no bones, none, 0. Its entire body is soft tissue, muscle, and nerve. The only hard structure in its entire frame is a small beak, shaped like a parrot's beak, hidden underneath where all 8 arms meet. That beak is the only part of its body that cannot compress, which means an octopus weighing over 30 pounds can squeeze through a hole the size of a quarter. If the i.e. beak fits, the entire animal fits. Aquarium workers have watched in disbelief as full-grown octopuses collapse their bodies like liquid and pour themselves through gaps no wider than a coin. And the blood running through those 3 hearts is not red. It is blue. Genuinely blue. Human blood uses iron to carry oxygen. That iron-based molecule, hemoglobin, is what gives our blood its red color. Octopuses evolved a completely different solution. Their blood uses copper instead of iron, a molecule called hemocyanin. Copper-based blood is far more efficient at transporting oxygen in cold, low-oxygen environments deep underwater. It is heavier, thicker, and moves more slowly than our blood, which is exactly why the octopus needs 3 hearts to push it through its body. But something even more bizarre lurks inside this strange biology. What you are about to hear next might be the reason scientists now consider this animal one of the most alien forms of life on our own planet. Octopus does not have 1 brain. It has 9. 1 central brain sits between its eyes, roughly where you would expect a brain to be. But each of its 8 arms contains its own cluster of neurons, its own processing center, its own ability to make decisions. Scientists have confirmed that a severed octopus arm, completely disconnected from the body, will still react to stimulation. It will still reach for food. It will still pull away from danger. For up to an hour after separation, that arm acts as if it has a mind of its own. Because in a very real sense, it does. Of the roughly 500 million neurons inside an octopus, more than 350 million are located in the arms, not in the central brain. That means over 2/3 of its thinking power is spread across its body. Imagine if your hands could taste, smell, make decisions, and solve problems without your head ever knowing about it. That is the daily reality for every octopus on Earth. This design is unlike anything else in the animal kingdom. It is so strange, so foreign to how intelligence works in mammals, birds, or even other sea creatures, that one leading researcher described it this way: studying the octopus is probably the closest we will ever come to meeting an intelligent alien without leaving our own planet. That is not poetry. That is a direct quote from a neuroscientist at the University of Washington, published in peer-reviewed research. And yet, for decades, we underestimated them. We called them simple. We grouped them with clams and snails because they are all technically mollusks. We assumed a creature without a backbone could never be truly smart. We were spectacularly wrong. Because what those 9 brains are capable of will challenge everything you think you know about intelligence. And the deeper we look, the stranger it gets. Here is a question that keeps neuroscientists awake at night. If an octopus has 9 separate processing centers, where exactly is the self? Where does the thinking happen? Is the octopus a single mind with 8 helpers, or is it 9 minds cooperating inside one body? Nobody knows for certain, and that uncertainty is what makes this creature so unsettling to study. In September of 2025, a landmark study published in Scientific Reports finally gave us hard numbers. Researchers filmed 25 wild octopuses across 6 different shallow water habitats and recorded nearly 7,000 individual arm movements. They catalogued 12 distinct types of arm actions. What they found shattered the assumption that all 8 arms work the same way. The front arms primarily handle exploring and investigating. The rear arms focus on pushing the body forward and steering through water. The middle arms do a bit of both. Each arm has a specialty. Each arm has a role. But here is where it gets truly unsettling. Despite these specializations, every single arm retains the full ability to do everything. Rear arm can explore if needed. A front arm can walk if required. The specialization is a preference, a habit, a choice. The arms are not locked into their roles. They choose them. That implies a level of coordination and flexibility that we are only beginning to understand. Think of it like a team of 9 highly skilled workers. The central brain acts as a manager, setting broad goals and priorities. Go find food, hide from that predator, investigate that shiny object. But once the command is issued, the arms take over. They figure out the details on their own. They decide how to grip, how to taste, how to move, how to react to unexpected obstacles. The central brain does not micromanage. It trusts its arms. This is called distributed intelligence, and it has enormous implications far beyond marine biology. Researchers working in artificial intelligence have started looking at the octopus as a blueprint for a completely new kind of computing. Right now, most of our technology runs on centralized systems. One powerful processor does all the work. But the octopus proves that intelligence can emerge from a network of smaller, semi-independent processors working together without constant oversight from a central controller. In 2025, the number of published scientific papers on octopus-inspired robotics grew by more than 50% compared to just 4 years earlier. Engineers are now building soft robots with flexible arms that process sensory information locally, just like an octopus does. The military, medical, and deep-sea exploration industries are all watching closely, because a robot that thinks the way an octopus thinks could go, places no human and no traditional machine ever could. But we need to pause here and appreciate just how weird this really is from a biological standpoint. In every intelligent animal we have studied before, intelligence lives in the head. Dolphins, crows, chimpanzees, elephants, humans. The brain calls the shots, and the body obeys. The octopus flipped that entire model on its head. Or rather, it spread its head across its entire body. There is a detail from laboratory research that perfectly captures how independent these arms truly are. Scientists at the University of Washington gave octopuses interesting objects to explore. Lego bricks, textured rocks, cinder blocks, elaborate mazes with food hidden inside. They watched as different arms simultaneously performed different tasks, completely unaware of what the other arms were doing. One arm would be solving a maze while another arm was tasting a rock on the opposite side of the tank. This raises a philosophical question that science has no answer to yet. Does the octopus experience this as one unified consciousness? Or does each arm have its own tiny sliver of awareness? When an arm tastes something new, does the octopus know it? Or does the arm know it first and only pass along the information if it seems important enough? We do not know. And what waits in the next chapter suggests the mystery only deepens when you look at what these arms can actually do. Picture this. You are a researcher in a marine laboratory. You place a sealed jar with a live crab inside on the floor of an octopus tank. The octopus has never seen a jar before. Within minutes, it wraps its arms around the glass and each arm begins working independently. One arm grips the base, another finds the lid, a third begins twisting. The octopus unscrews the jar, removes the lid, reaches inside, and pulls out the crab. The whole process takes less than 2 minutes. That experiment has been repeated in laboratories around the world, with the same result. Confirmed. Now here is what makes that truly remarkable. The octopus did not learn how jars work from watching someone open one. Nobody taught it. The arms figured it out through touch alone. Each sucker on an octopus arm is packed with chemoreceptors, the same type of sensory cells that exist on your tongue. An octopus does not just grip objects, it tastes them. Every single sucker on every single arm is simultaneously feeling the texture, the temperature, the chemical composition, and the shape of whatever it touches. A large octopus can have over 2,000 suckers across all 8 arms. Each sucker can move independently. Each one can grip with surprising force. A giant Pacific octopus, the largest species in the world, can lift roughly 30 pounds with a single sucker. Now multiply that across 2,000 suckers. The combined gripping power of a full-grown giant Pacific octopus is staggering. But raw strength is not what makes these arms extraordinary. It is the decision-making that happens inside them. Researchers have confirmed that the nerve clusters in each arm, called ganglia, can initiate action on their own. A sucker touches something interesting. The ganglion in that arm processes the information. If it is food, the arm moves toward the mouth. If it is a threat, the arm recoils. If it is something unfamiliar, the arm investigates further. All of this can happen before the central brain is even aware of it. In 2019, a model of information flow in octopus arms was presented at a neuroscience conference. It was the first comprehensive attempt to map how data moves between suckers, arms, and the central brain. The findings supported what many researchers had suspected. The suckers themselves can trigger coordinated actions with neighboring suckers, forming a chain reaction that moves up the arm before ever reaching the brain. Information flows from the bottom up, not the top down. Think about what that means for reaction time. A predator touches one arm. The arm reacts instantly, pulling away or releasing a cloud of ink before the brain even registers the threat. By the time the central brain catches up and says, "Danger," the arm has already handled it. This is parallel processing in a living animal, operating at a speed that centralized brains simply cannot match. And there is one more detail that researchers find deeply strange. An octopus arm does not just act on its own, it seems to be aware of its own position in space. In 2025, scientists tested octopuses with a version of the rubber hand illusion, a test originally designed for humans. In humans, if you hide someone's real hand and stroke a rubber hand in front of them at the same time, their brain starts to believe the rubber hand is their own. Researchers adapted this test for octopuses. The results showed that octopuses have a sense of body ownership, an awareness of where their arms are and what belongs to them. That is something scientists previously believed only existed in humans and a small number of mammals. The finding suggests octopuses do not just react, they perceive themselves. They have some form of self-awareness built into their arms. We still do not fully understand how this works, and that mystery is one of the great open questions in neuroscience today. But what we do know is that these tasting, thinking, self-aware arms are controlled by an animal that can do something even harder to explain. It can vanish. A A full-grown common octopus weighs about 22 pounds. It can be over 3 feet long with its arms stretched out, and it can flatten its entire body and slide through a gap barely an inch wide. If you have ever watched a video of this happening, you know it looks physically impossible, like a magic trick performed by a living animal. But it is pure biology, and the engineering behind it is extraordinary. Remember, an octopus has no skeleton, no bones, no cartilage, no rigid internal frame of any kind. The only hard part of its body is that parrot-like beak at the center of its arms. So when an octopus approaches a narrow opening, it first extends one arm through to test the space. If the beak can fit through, the rest of the body will follow. The key is in the muscle, structure. Octopus arms and body walls are built from what scientists call a muscular hydrostat. That is a fancy term for a muscle system that uses fluid pressure instead of bones for support. Your tongue is actually a muscular hydrostat too. It can extend, retract, twist, and change shape without any bones inside it. Now imagine that same principle applied to an entire body weighing 22 pounds. Every part of the octopus can elongate, compress, twist, or stiffen at will. When squeezing through a tight space, the octopus first elongates its body, stretching itself thin like toothpaste being pushed through a tube. The mantle, which is the large rounded section behind the head, flattens and compresses. The arms may go through first, pulling the rest of the body behind them. The entire process can take less than a minute, even through openings that seem absurdly small. This ability serves one critical purpose in the wild: survival. Octopuses are soft, protein-rich animals with no shell, no armor, and no spines. They are a favorite meal for moray eels, sharks, dolphins, and even large fish. Their primary defense is to disappear into crevices, coral formations, and rocky gaps that their predators cannot follow them into. An octopus that can squeeze through a 1-inch gap can escape a predator with a 6-inch jaw. That size difference is the margin between life and death on a coral reef. Aquarium workers have learned this the hard way. Octopuses in captivity are notorious for escaping their tanks. They find the smallest gaps in lids, filter openings, or pipe connections, and compress themselves through. One famous case involved an octopus at a New Zealand aquarium that repeatedly escaped its tank at night, crawled across the floor of the building, and squeezed itself down a drainage pipe that led directly to the ocean. By the time the staff figured out what was happening, the octopus was long gone. But here is something that takes this ability from impressive to almost unbelievable. They do not just squeeze through spaces, they evaluate them first. Researchers have observed octopuses approaching narrow gaps, extending a single arm to measure the opening, and then deciding whether to attempt the squeeze or find another route. This is spatial reasoning. The octopus is calculating whether its body will fit through an opening it has never seen before. That requires an understanding of its own body size, and the ability to compare that size to external objects. In other words, it is a form of planning. Some researchers believe this spatial intelligence is processed locally in the arms, not in the central brain. The arm measures the gap. The arm ganglia calculate whether the beak will fit, and the arm sends a green light back to the brain. Others believe the central brain is more involved than we think, coordinating the effort from above. This debate remains unresolved, but either way, the implication is the same. An animal with no bones is performing geometry in real time with nothing more than muscle and nerve. And yet, the ability to squeeze through tight spaces may not even be the most astonishing physical trick in the octopus toolkit. What comes next involves something that happens on the outside of its body. Something so fast, so precise, and so scientifically baffling that it forced researchers to rethink what we know about how any animal interacts with light. An octopus can change the color of its entire body in 0.3 of a second. Less than a blink of your eye. One moment it is reddish brown, sitting on a rock. A fraction of a second later, it is the exact same shade of pale grey as the sand beneath it. The transformation is so fast and so complete that even trained marine biologists can lose sight of an octopus from just a few feet away. The secret lies in millions of tiny structures embedded in the skin called chromatophores. Each chromatophore is a small elastic sac filled with pigment. The colours available are red, orange, yellow, brown, and black. Each sac is surrounded by a ring of tiny muscles. When those muscles contract, the sac stretches open and the pigment inside spreads across a wider area of skin. The colour becomes visible. When the muscles relax, the sac snaps shut and the pigment disappears. Open and closed, visible and invisible. This happens independently for each chromatophore, and an octopus has hundreds of thousands of them, covering its entire body. Imagine having hundreds of thousands of individual pixels on your skin, each one controlled by its own set of muscles, each one capable of displaying a different color at a different time. That is essentially what an octopus is working with. And it can control them with stunning precision. It can display patterns, stripes, spots, waves of color that ripple across its body like a living screen. It can make one half of its body one color and the other half a completely different color at the same time. But chromatophores are only the first layer. Beneath them sits a second layer of cells called iridophores. These are not pigment sacs, they are reflectors. Iridophores contain stacks of tiny mirror-like plates that reflect light at specific wavelengths. This is how an octopus produces colors that its pigment sacs cannot create on their own, like shimmering blues and greens. The iridophores bounce environmental light back through the chromatophore layer above, adding brightness and new colors to the mix. And beneath the iridophores lies a third layer, leucophores. These are broad-spectrum reflectors that scatter all wavelengths of light equally, creating a white backdrop. Think of them as a blank canvas underneath the two painting layers above. By adjusting the leucophores, the octopus controls the overall brightness and intensity of whatever pattern it is displaying. 3 layers working together. Pigment on top, mirrors in the middle, and a white reflector on the bottom. All controlled, in real time, all adjustable at the level of individual cells. Scientists have confirmed this multi-layer system through microscopy and direct observation, and it is considered one of the most sophisticated camouflage systems in the entire animal kingdom. But the real question is speed. How does the octopus coordinate all of this so fast? Scientists believe part of the answer lies in direct neural control. Chromatophores are wired directly to the nervous system. The octopus does not need to produce hormones or wait for chemical signals to travel through its blood. The command goes straight from nerve to muscle, and the color changes almost instantly. In laboratory experiments, researchers have observed octopuses adjusting their chromatophore patterns to match new backgrounds within 1 to 2 seconds of being placed on them. They can match the grain of sand. They can match the mottled texture of a rock covered in algae. They can even produce moving patterns that create a visual distraction, like a hypnotic wave rolling across their body, which they use to stun prey before striking. And remember, this is all happening across a body with no bones and no fixed shape. The skin stretches and compresses as the octopus moves, yet the pattern stays perfectly calibrated. The chromatophores adjust dynamically as the skin surface area changes. It is like having a high-definition screen printed on a water balloon. There is, however, a massive contradiction hiding inside this incredible system, and it is one of the most confusing mysteries in all of marine biology. Something that has baffled scientists for decades and still has no definitive answer. The animal performing this perfect color matching cannot see color at all. This is one of the strangest contradictions in nature. The greatest camouflage artist on the planet is completely colorblind. That is a confirmed scientific finding, and it has driven researchers to the edge of frustration for over 20 years. Here is the evidence. Scientists have dissected octopus eyes and examined the photoreceptor cells inside. In human eyes, we have 3 types of photoreceptors called cones, each sensitive to a different wavelength of light: red, green, and blue. Our brain combines the signals from all 3 to produce the full range of colors we see. Dogs have 2 types, which is why their color vision is limited. Octopuses have 1, a single type of photoreceptor. By every measure of visual biology, this means they can only detect differences in brightness, light and dark, black and white. They should be completely incapable of perceiving color, but they match colors flawlessly. Put an octopus on bright orange coral, and it turns bright orange. Move it to green algae, and it shifts to green. Place it on a checkerboard of black and white, and it will match that pattern perfectly. But here is the key detail from laboratory experiments. When researchers placed octopuses on checkerboards made of different colors that had the same brightness level, like yellow and blue of equal lightness, the octopuses could not distinguish between them. They failed the test. They could not tell the colors apart when brightness was equal. This strongly supports the conclusion that they truly are colorblind. So how on earth do they match colors they cannot see? This mystery has generated several competing theories, and none of them has been proven beyond doubt. The first theory involves their bizarre pupils. Unlike humans whose pupils are round, octopus pupils are horizontal slits. Cuttlefish, their close relatives, have W-shaped pupils. Squid have dumbbell-shaped pupils. In 2016, a father and son research team from the University of California, Berkeley, and Harvard University proposed that these unusual pupil shapes might allow cephalopods to exploit a phenomenon called chromatic aberration. When light passes through any lens, different colors bend at slightly different angles. Normally, eyes are designed to minimize this effect so the image stays sharp. But the researchers argued that cephalopod eyes might be doing the opposite. By deliberately allowing chromatic aberration, the octopus could determine the color of an object based on how blurry or sharp it appears at different focal distances. It is an entirely different method of detecting color, one that no other animal uses. This theory fits some of the data. Cephalopod lenses do show more chromatic aberration than most other eyes, and the odd pupil shapes would amplify the effect. But it has not been proven in live animals yet. It remains a leading hypothesis, not a confirmed fact. The second theory is even stranger. What if the octopus can see color through its skin? In 2015, researchers discovered that octopus skin contains opsins, light-sensitive proteins normally found in eyes. Laboratory experiments showed that isolated octopus skin, disconnected from the brain and the eyes, responded to changes in light. Chromatophores expanded when exposed to light. This means the skin itself can detect light independently. Whether this ability extends to detecting color remains unknown, but if it does, the octopus would essentially have a second visual system spread across its entire body. A third theory focuses on polarized light. Unlike humans, octopuses can detect the polarization of light, meaning the direction in which light waves vibrate. Different materials reflect polarized light differently. It is possible that octopuses use polarization cues to infer what colors surround them, even without directly seeing those colors. This theory has less supporting evidence than the other two, but it has not been ruled out. The honest answer is that we do not yet know how octopuses match colors they cannot see. It is one of the biggest unsolved mysteries in animal biology, and the fact that we cannot answer this question after decades of research tells you something profound about how different this animal is from everything else we have studied. What we do know is that their skin is doing something no other animal's skin does. And the next discovery about that skin pushed the mystery into even stranger territory. In 2015, a team of researchers from the University of California, Santa Barbara, confirmed something that sounded like science fiction. Octopus skin contains the same light-sensitive proteins that exist inside eyes. The protein is called rhodopsin, and in virtually every other animal on Earth, it is found only in the retina. Finding it scattered across the skin of an octopus was unexpected and deeply puzzling. The researchers tested this by shining light directly onto patches of isolated octopus skin. No eyes involved, no brain connected, just skin in a dish. The chromatophores responded. They expanded when light hit them. They changed the pigment display. The skin was reacting to light completely on its own. This finding has been replicated and is considered confirmed by the scientific community. But what exactly does this mean? Can the skin see in the way that eyes see? Almost certainly not. The skin has no lens to focus light. It has no retina to form an image. There is no neural wiring in the skin sophisticated enough to assemble a picture of the world. What the skin can do, at minimum, is detect changes in brightness. It knows when light hits it and when it does not. That alone could be incredibly useful for camouflage. If a shadow falls across one section of the body, that section could darken its chromatophores locally without needing a signal from the brain or the eyes. Some scientists speculate the ability goes further. If the skin contains light-sensitive proteins that respond to specific wavelengths, it might be able to detect color at a very crude level. Enough to know the general color of the background without needing the eyes to relay that information. This would solve the colorblind camouflage paradox in a radically unexpected way. The octopus would not be seeing color with its eyes, it would be sensing it with its entire body. That theory remains unproven. Experiments have shown the skin responds to light intensity, but convincing evidence that it responds to specific colors has not yet been published. The possibility remains open, and multiple labs around the world are actively investigating it. Here is what makes this finding so significant beyond just octopus biology. If it turns out that a complex organism can distribute visual sensing across its entire body surface, it would upend a basic assumption in neuroscience. We have always believed that seeing requires eyes, dedicated organs with lenses and retinas connected to a brain that processes visual information. The octopus may be proving that evolution found another way, a way to put a primitive version of sight directly into the skin, bypassing the eyes entirely. There is another layer to this mystery. Researchers have observed that octopuses can adjust their camouflage even when their eyes are covered. In some experiments, octopuses that could not see their surroundings still made partial color adjustments. The adjustments were not as accurate as when the eyes were uncovered, but they were better than random chance. Something was helping the animal sense its environment without using its eyes. The most likely explanation is the light-sensing skin. Consider the implications. Every inch of the octopus body is simultaneously processing information about the light environment. The eyes scan for predators. The skin monitors the brightness of the background. The arms taste the chemistry of whatever they touch. The ganglia in each arm make local decisions, and the central brain coordinates all of it, blending inputs from hundreds of thousands of sensors into one seamless response. It is a full-body sensory network, unlike anything else in the animal kingdom. We should note that much of this remains in the early stages of research. The 2015 discovery opened a door, but we are still walking through it. Claims about octopus skin seeing color should be treated as promising theory, not established fact. The skin detects light. That is confirmed. Whether it detects color is the question that could change everything. And speaking of questions that could change everything, there is one part of the octopus eye that hides a secret so elegant, so clever, that it might make the colorblind label meaningless altogether. Look into the eye of an octopus, and you will see something genuinely alien. The pupil is not round like a human's. It is not vertical like a cat's. It is a horizontal slit, sometimes slightly curved, stretching across the eye like a thin, dark line. In their close relatives, the cuttlefish, the pupil is shaped like the letter W. In some squid, it looks like a dumbbell. These are the strangest pupil shapes in the entire animal kingdom, and scientists believe they may hold the key to a sensory trick that rewrites the rules of vision. Here is the conventional understanding. Round pupils, like ours, are designed to shrink down to tiny circles in bright light. When the pupil gets very small, it acts like a pinhole camera, forcing all wavelengths of light to focus on the same point on the retina. This eliminates chromatic aberration, the blurring that occurs because different colors of light bend at different angles when passing through a lens. Eliminating chromatic aberration gives us sharper, clearer images. That is how mammalian vision works. The 2016 hypothesis from Alexander Stubbs and Christopher Stubbs proposed that cephalopods are doing the exact opposite. Instead of eliminating chromatic aberration, they are deliberately using it. Their wide, slit-shaped pupils allow light to enter the eye from many different angles. This maximizes chromatic aberration. Different colors of light focus at different distances from the retina. A blue object would produce a sharp image at one focal depth and a blurry image at another. A red object would do the reverse. By sweeping its focus back and forth, or by comparing the sharpness of images at different focal points, the octopus could theoretically figure out what color an object is. A sharp image means the object matches the current focal distance. A blurry image means it does not. The color that produces the sharpest image at a given setting would reveal the color of the object. If this is true, the octopus is using physics to see color with only one type of photoreceptor. No animal has ever been confirmed to do this. It would be a completely unique form of color vision, different from the receptor-based system used by every other creature with color sight. The researchers tested this idea using computer simulations based on the known optics of cephalopod eyes. The results were promising. The simulations showed that slit-shaped and W-shaped pupils would produce enough chromatic aberration to allow meaningful color discrimination, especially for objects against a contrasting background. Brightly colored objects on a reef would produce noticeably different focus patterns than dull gray objects. The system would work best in well-lit shallow water, which is exactly where many octopuses and cuttlefish live. There is additional supporting evidence. Scientists have known for decades that cephalopod lenses do not correct for chromatic aberration the way mammalian lenses do. Previously, this was considered a flaw. The Stubbs hypothesis reframes it as a feature. Evolution did not fail to correct the aberration. It kept it on purpose. However, this has not been directly tested on living animals. No experiment has yet demonstrated that a live octopus actually uses chromatic aberration to determine color. The theory is elegant and consistent with anatomical data, but it remains a hypothesis, not a proven mechanism. The scientific community is waiting for behavioral experiments that could settle the question. If researchers can show that octopuses with artificially restricted pupils lose the ability to match background colors, that would be strong evidence in favor of the chromatic aberration theory. Until then, it sits alongside the skin sensing theory and the polarization theory as one of several possible explanations for the colorblind camouflage mystery. What is clear is that the octopus eye, despite looking remarkably similar to a human eye from the outside, works in a fundamentally different way. Our eyes evolved to suppress optical distortions. Theirs may have evolved to exploit them. And that eye belongs to an animal that does something with its entire body that no other creature on Earth can match. Something so outrageous that when it was first filmed, scientists did not believe what they were watching. In 1998, on a muddy ocean floor off the coast of Sulawesi, Indonesia, scientists discovered a creature that threw everything they knew about animal camouflage into chaos. It was an octopus, about 2 feet long, with thin striped arms and a small body. What made it different from every other octopus ever documented was what it did when it felt threatened. It did not just change color, it changed what it was. Within seconds, this animal contorted its body, rearranged its arms, and altered its swimming style to impersonate a completely different species. When confronted by aggressive damselfish, it tucked 6 arms close to its body, extended 2 behind it in opposite directions, and undulated through the water like a banded sea snake. When a predator approached from below, it spread its arms wide and flattened its body to mimic the broad, flat shape of a venomous lionfish. When it wanted to move across open ground, it pressed its body flat against the sand and rippled along the bottom like a toxic flatfish. This was the mimic octopus, and since its discovery, researchers have documented it impersonating at least 15 different marine species: lionfish, flatfish, sea snakes, jellyfish, mantis shrimp, stingrays. Each impersonation involves a specific body posture, a matching color pattern, and a convincing imitation of the other animal's movement style. It is the most elaborate mimicry ever observed in any animal on the planet, confirmed across multiple peer-reviewed studies. What makes this even more astonishing is the evidence that the mimic octopus selects which animal to impersonate based on the specific threat it faces. When attacked by damselfish, it chose to mimic a banded sea snake, one of the damselfish's primary predators. It did not randomly pick an animal, it picked the right one. That suggests the octopus is identifying its attacker, assessing the threat, recalling which impersonation would be most effective, and executing a full-body transformation in seconds. That is strategy. That is decision-making under pressure. That is intelligence of a kind we still struggle to explain in an invertebrate. Most animals that use mimicry are locked into one disguise. A hoverfly looks like a wasp. A viceroy butterfly resembles a monarch. They are born with their disguise and cannot change it. The mimic octopus can switch between disguises on the fly, selecting the most appropriate one for each situation. No other animal does this, and the mimic octopus is not the only species pulling off extraordinary deceptions. The common octopus can match almost any background on a coral reef in less than a second. The wonderpoos, a close relative of the mimic, has its own repertoire of visual tricks. The algae octopus can extend its arms, raise its skin into bumpy textures, and sway gently back and forth, becoming virtually indistinguishable from a clump of seaweed. This is so convincing that small fish have been filmed swimming right past the disguised octopus without noticing it. But here is a detail that many people miss. Camouflage in octopuses is not just defensive. They use it to hunt. An octopus can hide in plain sight on a coral head, waiting for an unsuspecting crab to walk within arm's reach. Some species use a more aggressive approach. They will produce a rapid-fire series of dramatic color changes, flashing bright patterns across their body so quickly that the prey freezes in confusion. In that split second of hesitation, the octopus strikes. Scientists call this a startling display, and it works because the sudden burst of pattern overwhelms the prey's visual processing. The crab's brain locks up for just long enough. Think about what all of this requires at a neural level. Pattern selection, real-time body reshaping, behavioral imitation, threat assessment, prey manipulation, all performed by an animal with a lifespan of 1 to 2 years that receives no teaching from its parents. Octopus mothers die before their young hatch. Every octopus that has ever lived learned these skills entirely, on its own. And the most resourceful species took the concept of improvisation even further. Because some octopuses are not just hiding and pretending to be other animals, they are picking up objects, carrying them around, and using them as tools. In 2009, on a sandy ocean floor off the coast of Indonesia, researchers from the Melbourne Museum in Australia filmed something that had never been documented in any invertebrate animal. A small octopus, about the size of a human hand, was walking across the sand carrying 2 halved coconut shells tucked underneath its body. When a potential threat appeared, the octopus stopped, flipped the shells around itself, and sealed itself inside like a living walnut. This was the coconut octopus, And what the researchers filmed was true tool use. The distinction matters. Many animals use objects in their environment for immediate benefit. A hermit crab crawls into a shell and lives there permanently. That is not tool use. Tool use, by scientific definition, requires an animal to collect an object, transport it, and deploy it for a specific purpose at a later time. The coconut octopus was doing exactly that. It was picking up shells, cleaning them with jets of water, carrying them across the seafloor for distances of over 60 feet, and then assembling them into a protective shelter only when needed. Carrying the shells was not easy. With half its arms occupied holding two large shell halves against its body, the octopus had to walk on its remaining arms in a slow, awkward, stilted gait. This movement was clumsy and exposed the animal to predators. It was a clear short-term cost for a future benefit. That kind of planning, accepting a present disadvantage for a later reward, is one of the hallmarks of advanced cognition. Before this discovery, tool use in the ocean was thought to be limited to marine mammals, like dolphins, which have been observed using sponges to protect their snouts while foraging on rocky seafloors. Finding the same behavior in a mollusk, an animal more closely related to clams and snails than to any mammal, sent shockwaves through the scientific community. The paper was published in the journal Current Biology and became one of the most widely cited studies in cephalopod research. Since that initial discovery, researchers have documented other species of octopus using objects in inventive ways. Some species stack rocks and shells to build barricades at the entrance of their dens. Others arrange debris in specific patterns around their homes. And one observation went even further into the unexpected. The blanket octopus, a species we will encounter in a later chapter, has been filmed ripping venomous tentacles of Portuguese man-of-war jellyfish and carrying them as weapons. Let that sink in. An octopus that is immune to the venom of one of the most feared creatures in the ocean deliberately tears off pieces of that creature and brandishes them at threats. That is not just tool use, that is weapon acquisition. Back on the sandy ocean floor, the coconut octopus added more evidence. Researchers noted that individual octopuses showed preferences for specific shell sizes and types. Some preferred coconut halves, others chose clamshells. This variability suggests individual decision-making and the ability to evaluate objects for fitness of purpose. Here is what makes tool use such a powerful indicator of intelligence. It requires the animal to imagine a problem that does not exist yet. The coconut octopus is not being attacked when it picks up a shell. It is anticipating a future scenario and preparing for it. That requires selecting an appropriate response in advance and taking physical action based on that prediction. This kind of thinking was once considered unique to humans, then extended to apes, then to crows and dolphins, and now to an invertebrate on a tropical ocean floor. The coconut octopus also demonstrated something else. It engaged in bipedal locomotion, walking on two arms while holding objects with the other six. This was one of the first confirmed cases of an octopus walking on two limbs, and it raised fresh questions about motor planning and coordination in animals without a backbone. And if tool use was the discovery that forced scientists to take octopus intelligence seriously, the next batch of observations from laboratories around the world proved it was just the beginning. Because octopuses in captivity started doing things that nobody could explain. In 2020, a research team studying octopus hunting behavior in the Red Sea captured footage of something no one expected. An octopus was swimming alongside a group of fish. They were hunting cooperatively, as octopuses and certain fish species sometimes do, flushing prey out of coral crevices. The octopus would reach into a gap, and the fish would wait on the other side to catch whatever fled. A working partnership. Then, without any obvious provocation, the octopus hauled off and punched one of the fish. Not a gentle nudge, a sharp, explosive strike with one arm. The fish tumbled sideways in the water. The octopus continued hunting as if nothing had happened. Researchers watched this behavior over and over during their observation period. Octopuses punched their fish partners repeatedly, under different circumstances, and the scientists identified two possible explanations. The first was practical. Sometimes a fish would try to steal food that the octopus had flushed from a crevice. The punch seemed to be a punishment, a way of enforcing cooperation and discouraging cheating. You take my food, you get hit. But the second explanation was the one that grabbed headlines around the world. In some cases, the punch appeared to have no immediate practical benefit. The fish had not stolen anything. It was not in the way. The octopus just punched it. The researchers considered the possibility that some of these punches were, scientifically speaking, spiteful. The octopus was lashing out because it could. Because the fish annoyed it. Because it felt like it. This study was published in the journal Ecological and became one of the most discussed papers in animal behavior that year. The idea that an octopus could act out of frustration or spite implies emotional states more complex than simple survival instincts. We cannot prove that octopuses feel spite the way humans do. That remains an open question. But the behavior pattern is difficult to explain through purely rational survival logic. And this is just one example of octopus behavior that hints at something deeper than basic instinct. In aquariums around the world, octopuses have demonstrated a range of behaviors that challenge the traditional boundaries between animal and intelligent being. They have been documented playing with objects. Marine biologist Roland Anderson observed octopuses repeatedly pushing bottles across their tanks, bouncing them, off the walls, catching them, and pushing them again. There was no food reward, no survival value. The octopuses appeared to be entertaining themselves. Play behavior is considered a strong indicator of cognitive complexity because it requires an animal to engage with the world in a way that has no immediate practical purpose. The animal is exploring possibilities, testing boundaries, having fun. Play has been documented in mammals and birds, but finding it in a mollusk was extraordinary. Octopuses have also been observed solving mechanical puzzles in captivity. Researchers present them with boxes that have multiple locking mechanisms, latches, and sliding panels that must be operated in the correct sequence to access food inside. Octopuses figure these out, often within minutes, and they remember the solution. When presented with the same puzzle weeks later, they solve it faster than the first time. That is long-term memory in action. One particularly famous case involved an octopus at the University of Otago in New Zealand that learned how to short-circuit the aquarium lights. The overhead lighting apparently annoyed the animal. It aimed a jet of water at the bulb, caused an electrical short, and turned off the light. It did this so many times that the aquarium released it back into the ocean because the repairs were too expensive. And then there are the escapes. An octopus at the National Aquarium of New Zealand made international news when security cameras revealed it had been climbing out of its tank at night, crawling across the wet floor, entering a neighbouring tank to eat the fish inside, and then crawling back to its own tank before the staff arrived in the morning. It did this repeatedly without being caught, until a trail of water on the floor gave it away. These are not instinctive behaviours. These are creative solutions to novel problems, and they reveal something about octopus cognition that the next chapter explores in even more unsettling detail. Let us be clear about something. When we say octopuses escape from tanks, we are not talking about an animal accidentally finding a gap and slipping through. We are talking about systematic, repeated, and increasingly clever efforts to defeat the security measures that human engineers put in place, specifically to contain them. Aquariums around the world have invested significant resources into octopus-proofing their enclosures. Lids are weighted down. Gaps are sealed with duct tape and cable ties. Water intake pipes are covered with mesh, and octopuses still find ways out. They test every edge, every seal, every connection point. They probe with their arms, tasting and measuring the entire perimeter of their enclosure until they find a weakness. Then they exploit it. One documented case from a marine research facility describes an octopus that figured out how to unscrew the bolts on the inside of its tank lid. The staff switched to a different fastening system. The octopus figured that one out too. They added a second layer of security. The octopus defeated it within a week. This back and forth between human engineers and a single octopus lasted months. What makes these escapes scientifically significant is the evidence they provide for multiple cognitive abilities working together. First, the octopus must explore its environment and build a mental map of the tank, including all potential exit points. That requires spatial memory. Second, it must identify which exit points are vulnerable. That requires evaluation and judgment. Third, it must manipulate physical objects like lids, bolts, and mesh to create an opening. That requires dexterity and problem solving. Fourth, many escapes happen at night when the lights are off and staff are absent. That suggests the octopus understands, at some level, the difference between being observed and being unobserved. That implies a form of timing, if not outright planning around human schedules. The case of the fish raiding octopus is particularly telling. This was an octopus that not only escaped its own tank, traveled across the floor and entered another tank to hunt, but also returned to its own tank afterward and closed the lid behind it. Think about the cognitive chain involved. The octopus had to remember the location of the food source. It had to navigate an unfamiliar environment in the dark. It had to enter the second tank, hunt successfully, and then reverse the entire process. And it had to do all of this while managing a body that was out of water, breathing through gills that were not submerged, on a time limit before dehydration became dangerous. Octopuses can survive outside of water for short periods because their gills can extract oxygen from moist air. But this ability has limits. The fact that the octopus successfully completed the round trip multiple nights in a row means it was managing its time outside the water with some understanding of its own physical limitations. This is also where enrichment programs become important. Many modern aquariums have realized that keeping octopuses mentally stimulated is not optional. A bored octopus becomes a destructive octopus. It will damage its tank, harass neighboring animals, and spend its energy trying to escape. Enrichment programs provide puzzles, toys, mazes, and novel objects to keep the animal engaged. Researchers at multiple institutions have confirmed that octopuses in enriched environments show fewer escape attempts and more engaged, investigative behavior. Some aquariums have gone even further. They rotate toys and puzzles on a schedule to prevent the octopus from losing interest. They introduce new textures, new colors, and new challenges regularly. One facility reported that their octopus had a clear preference for certain puzzle types and would ignore puzzles it considered too easy. Let that settle for a moment. An animal with a lifespan of 1 to 2 years, no parental teaching, no social learning from other octopuses, and a brain smaller than a walnut can get bored with puzzles it considers beneath it. And yet, the strangest part of octopus intelligence in captivity is not the escapes, the problem solving, or even the play. It is something far more personal. Here is a fact that unsettles many people when they first hear it. Octopuses can recognize individual human faces. They respond differently to different people, and they remember who treated them well and who did not. This has been documented across multiple aquarium facilities and marine research laboratories worldwide. Staff members who regularly feed and interact gently with octopuses report that the animals approach them, extend arms toward them, and make what researchers describe as affiliative contact. Gentle touching, arm holding, investigative probing that is calm and exploratory rather than defensive. Staff members who have handled octopuses roughly, performed medical procedures, or simply annoyed them receive a very different greeting. The octopus will track them with its eyes as they walk past the tank, and then, with startling accuracy, It will aim a jet of water directly at that specific person, not at the person standing next to them. Not randomly, at them. One widely cited case from the University of Otago in New Zealand involved an octopus that consistently squirted water at one particular staff member every time she walked past. Other staff could approach the tank without incident, but as soon as this specific person appeared, the octopus would position itself at the edge of the tank and fire. The behavior was so targeted and so consistent that it became a running joke at the facility. But underneath the humor was a serious scientific implication. The octopus was demonstrating individual recognition, long-term memory, and what appeared to be a sustained grudge. Individual recognition is a cognitively demanding task. It requires the animal to process and remember unique visual features of a specific face or body, store that information over time, associate it with either positive or negative experiences, and then retrieve that association when the individual reappears. This ability is well documented in mammals and some birds. Finding it in a mollusk was, once again, a surprise. But wait, octopuses are colorblind and live underwater, where visibility is often poor. How are they identifying individual humans? The leading theory is that they use a combination of visual cues beyond color, including body shape, movement patterns, and possibly even the chemical traces that different humans leave near the tank. Remember, every sucker on every arm can taste the surrounding water. An octopus may be identifying people partly by their chemical signature, a biological version of recognizing someone by their scent. There is also evidence of more complex social responses. Some captive octopuses have been observed engaging in what keepers describe as playful interactions with familiar humans. They will gently hold a keeper's hand and explore their fingers with their suckers. They will accept food delicately from one person while snatching it aggressively from another. They will follow a trusted keeper's movements around the room with their eyes, tracking them with what appears to be curiosity rather than fear. These observations are difficult to quantify and study rigorously because they involve subjective interpretations of behavior. Is the octopus really being playful, or is it simply investigating a familiar stimulus? Is it holding your hand because it trusts you, or because your hand is warm and chemically interesting? Scientists are cautious about attributing human-like emotions to non-human animals, and rightly so. But the consistency of these reports across dozens of facilities and hundreds of individual octopuses makes it hard to dismiss entirely. And here's an important note about personality. Researchers have confirmed that individual octopuses within the same species show distinct personality traits. Some are bold and exploratory. Some are shy and hide at the first sign of a new stimulus. Some are aggressive. Some are gentle. Some are curious. Some are indifferent. This individual variation in temperament has been measured using standardized behavioral tests and is considered a genuine finding, not just anecdotal observation. An animal that recognizes faces, holds grudges, has individual personality, and engages in playful behavior is an animal that is pushing the boundary of what we consider consciousness. And that boundary got even blurrier when researchers observed octopuses doing something that, until recently, was believed to be limited to the most cognitively advanced animals on Earth. There is a simple test that scientists use to separate complex minds from simple ones. Can the animal play? Play is defined in behavioral science as a voluntary activity that has no immediate survival value. It is not hunting. It is not mating. It is not defending territory. It is doing something just for the experience of doing it. Finding play in an animal tells you that its brain has spare capacity beyond pure survival, and that it is motivated by curiosity or enjoyment rather than just instinct. Dolphins play, crows play, parrots play, elephants play, and octopuses play. Marine biologist Roland Anderson at the Seattle Aquarium conducted one of the first systematic studies of play behavior in octopuses. He gave giant Pacific octopuses small floating bottles in their tanks. No food was inside. No reward was attached. The octopuses picked up the bottles, examined them, and then began pushing them back and forth across the tank. They aimed the bottles at the water intake jet so that the current would push the bottles back to them, creating a kind of catch game with the tank's own water flow. They did this repeatedly, session after session, without any food reward or external motivation. Anderson documented that different octopuses played with the bottles in different ways. Some pushed gently and waited for the return. Others slammed the bottles hard against the glass. Some appeared to lose interest quickly, while others played for extended periods. This individual variation in playstyle further supports the argument for personality in octopuses. Other facilities have reported similar findings. Octopuses given Lego bricks will manipulate them, stack them, and rearrange them. One researcher described an octopus that would repeatedly pull a small floating toy underwater, release it, watch it bob back to the top, and then pull it down again, over and over, like a child with a bath toy. Play behavior has also been observed in wild octopuses, though documenting it in natural settings is harder. Divers have reported seeing octopuses manipulating shells in ways that appear to have no functional purpose, flipping them, stacking them, tossing them short distances. These accounts align with the controlled laboratory findings. Why does play matter so much in the context of intelligence? Because play requires several cognitive abilities working together. The animal must be free from immediate survival pressures, meaning it has enough food, enough safety, and enough environmental stability to devote energy to non-essential activities. It must be able to evaluate objects for interesting properties beyond just food or danger. It must maintain attention on an activity that has no tangible reward. And in some cases, it must invent novel uses for objects, which is a form of creativity. The existence of play in octopuses poses a challenge to theories of intelligence that rely on social complexity. The social intelligence hypothesis argues that big brains evolved because animals needed to navigate complex social relationships within their groups. This works well for dolphins, primates, and elephants, but octopuses are loners. Most species are solitary their entire lives. They have no social hierarchy to navigate. This has led some researchers to propose an alternative called the ecological intelligence hypothesis. It argues that octopuses evolved their impressive brains to navigate their complex and dangerous physical environment. A coral reef is a three-dimensional maze filled with hiding spots, ambush predators, shifting currents, and thousands of potential prey items that each require a different hunting strategy. Surviving in that environment as a soft, boneless animal with no shell requires constant problem-solving, rapid learning, and creative improvisation. Play may be a byproduct of that ecological intelligence. An octopus brain built for solving complex physical problems in a dangerous environment may simply have enough cognitive overhead to engage with the world in a non-survival context. Play, in this view, is what intelligence does when it has nothing urgent to deal with. And that intelligence belongs to an animal kingdom that contains creatures far stranger than the common octopus. What lies ahead involves a species so extreme in its biology that it makes every other octopus look ordinary by comparison. Somewhere in the open tropical ocean, far from any reef or coastline, drifts one of the most visually stunning creatures alive. The female blanket octopus unfurls a sheet of iridescent membrane between her arms that flows behind her like a living cape. She can stretch over 6 feet long, shimmering with silvers and purples as she moves through the water. She is large, powerful, and striking. Now picture the male. He is smaller than your thumbnail, less than 1 inch long. When scientists first discovered the male blanket octopus, they did not believe it was the same species. The size difference between the male and female is roughly 10,000 to 1 by weight. This is one of the most extreme cases of sexual size dimorphism in the entire animal kingdom. Nothing else comes close. Not anglerfish, not spiders, not any other known animal. Why is the difference so dramatic? The answer lies in their reproductive strategy and the open ocean environment where they live. In the deep blue water, there is no reef to hide behind, no rocks to crawl into, no coral to use as camouflage. A small male has almost no defense against predators. His survival strategy is to stay tiny, nearly invisible, and to find a female as fast as possible. He does not need to grow large because he only needs to mate once. After transferring his sperm, the male detaches one of his specialized arms, called a hectocotylus, and gives it to the female. That arm, loaded with reproductive material, is stored by the female until she's ready to fertilize her eggs. The male, meanwhile, drifts away and dies shortly after. The female, on the other hand, benefits from being large. Her size allows her to carry more eggs, travel farther in search of food, and survive the hazards of open ocean life. Her flowing membrane serves multiple purposes. When threatened, she unfurls it to make herself appear much larger than she actually is. The billowing sheet of skin can also be detached in pieces, leaving behind a floating decoy that confuses a predator while she escapes. But the blanket octopus's most remarkable trick has nothing to do with her size. It has to do with how she arms herself, and it is one of the most creative defensive strategies in the ocean. Young blanket octopuses have been observed tearing tentacles from Portuguese man-of-war, one of the most venomous creatures in the sea. The man-of-war's tentacles are loaded with thousands of microscopic stinging cells called nematocysts. Contact with human skin causes excruciating pain. In severe cases, it can cause cardiac arrest. The blanket octopus is immune to this venom, so it rips the tentacles off and carries them around like loaded weapons, wielding them at anything that threatens it. This behavior has been documented by marine researchers and is considered confirmed. A small, vulnerable octopus with no venom of its own, no shell, and no size advantage deliberately acquires the most painful weapon available in its environment and uses it for defense. That is not instinct in any simple sense. That is resourcefulness on a level that most animals never approach. The blanket octopus also hunts using these stolen tentacles, wrapping them around prey to paralyze it before feeding. The venom does the work. The octopus just delivers it. This species has been difficult to study because it lives in the open ocean, far from shore, and is rarely encountered. Most of what we know comes from chance encounters by divers and a handful of dedicated research expeditions. But every observation adds to the picture of an animal that compensates for its vulnerability with ingenuity. The blanket octopus is proof that intelligence and creativity are not limited to octopuses living on coral reefs. Even in the featureless open water, where there are no objects to manipulate and no terrain to hide behind, these animals find solutions. And out in those same warm tropical waters lives another octopus species that took a different evolutionary path entirely. Instead of growing large or stealing weapons, it became one of the most dangerous animals on the planet through chemistry alone. Pick it up and you might die. That is the simplest warning anyone can give about the blue-ringed octopus. It fits in the palm of your hand. Its body, including the arms, spans barely 5 to 8 inches. Its normal color is a dull yellowish brown, almost invisible against the sandy rubble and rock crevices where it lives in shallow waters across the Indo-Pacific. But when this animal feels threatened, its skin erupts with dozens of brilliant neon blue rings. Those rings are a warning. One of the clearest and most honest warnings in the entire animal kingdom. Because inside the salivary glands of this tiny creature sits a toxin called tetrodotoxin, a neurotoxin so powerful that a single octopus carries enough to kill 26 adult humans. Tetrodotoxin is 1,200 times more toxic than cyanide. There is no antivenom. There is no antidote. If enough toxin enters your bloodstream, it blocks sodium channels in your nerves, shutting down your ability to send signals from your brain to your muscles. You become paralyzed. You stop breathing. Your heart may stop. Death can occur within minutes of a bite. The only treatment is immediate mechanical ventilation, keeping the victim breathing artificially until the toxin wears off. which can take up to 24 hours. Here is a strange detail. The octopus does not produce this toxin itself. The tetrodotoxin is manufactured by symbiotic bacteria that live inside the octopus's salivary glands. The octopus provides a home for the bacteria, and the bacteria provide the octopus with one of the deadliest weapons in nature. This arrangement is an example of symbiosis, a partnership between two organisms that benefits both. Scientists have confirmed that the same tetrodotoxin-producing bacteria are found in pufferfish, certain species of newts, and several other unrelated animals. The toxin itself is ancient and widespread in nature, but the delivery system varies from species to species. The blue-ringed octopus delivers it through a bite. In practice, bites on humans are extremely rare. The blue-ringed octopus is not aggressive. It is shy, reclusive, and prefers to hide. Most bites occur when someone accidentally steps on one in shallow water or, more commonly, picks one up out of curiosity without realizing what they are holding. The octopus typically tries to camouflage and flee before resorting to biting. The flashing blue rings are a last resort warning, the animal's way of saying, "I am deadly, and you should leave." During normal hunting, the blue-ringed octopus uses a much smaller dose of venom to subdue its prey. It feeds on small crabs, shrimp, and fish, biting them and injecting just enough toxin to paralyze them. It then uses its beak to break into the prey's shell or body and its digestive enzymes to soften the tissue for consumption. The precision is remarkable. Studies have shown that octopuses know exactly where to bite each type of prey to deliver the venom most effectively with the least effort. There are 4 recognized species in the blue-ringed octopus group, all small, all venomous, and all stunningly beautiful when their rings activate. They are daytime hunters, which is unusual for octopuses. Most species hunt at night. The blue-ringed octopus moves through shallow reef rubble during the day, relying on its camouflage to stay hidden. And here is one final detail that links this tiny killer back to the broader story of octopus intelligence. Blue-ringed octopuses have been observed using shelter strategies similar to the coconut octopus. They carry shells and small objects for protection. They evaluate hiding spots before committing to them. Despite their lethal venom, they still rely on intelligence and planning as primary survival tools. The venom is a backup, not a crutch. All octopuses carry venom, every single species. But in most of them, the venom is mild enough to pose no danger to humans. The blue-ringed octopus is the extreme outlier, a miniature animal armed with a chemical weapon out of all proportion to its size. And that weapon comes from bacteria, not from the octopus itself. But venom is just one piece of the octopus chemical toolkit, because there is another substance they produce that does something far more creative than killing. When most people think of an octopus squirting ink, they picture a dark cloud in the water, like a smoke screen. The octopus disappears behind it, and the predator loses sight of his prey. Simple. Effective. Case closed. But the reality is far more sophisticated than that, and the ink itself is doing things that scientists did not fully appreciate until recently. Octopus ink is produced in a specialized organ called the ink sac, located near the gut. The ink is a mixture of melanin, the same pigment that gives human skin its color, and mucus. When ejected, the ink can take several different forms, depending on the situation. Sometimes the octopus releases a diffuse cloud that spreads through the water, creating a visual screen. Other times, it ejects a thick, compact blob that holds its shape. That blob is roughly the same size and shape as the octopus itself. It is a decoy, a fake octopus made of ink. The predator lunges at the decoy, thinking it is the real animal. Meanwhile, the actual octopus has already changed color, flattened itself against the bottom, and slipped away in the opposite direction. Some species can produce multiple decoys in rapid succession, leaving a trail of phantom octopuses behind them as they flee. Researchers have observed individual octopuses creating as many as 5 or 6 decoys in a single escape sequence, each one drifting in a slightly different direction. But the ink does more than fool the eyes. Research has shown that octopus ink contains chemicals that interfere with a predator's sense of smell and taste. Many ocean predators, including moray eels and some shark species, rely heavily on chemical cues to track prey. The melanin and mucus in octopus ink coat the predator's chemoreceptors, temporarily blinding its sense of smell. It is a chemical weapon against the nose. There is also evidence that certain compounds in the ink irritate the gills of fish predators, causing discomfort and discouraging pursuit. The combined effect is devastating. A predator that encounters octopus ink loses its sight to the cloud, loses its sense of smell to the chemicals, and feels physical irritation in its gills. By the time it recovers, the octopus is long gone. Different species deploy their ink in different ways, depending on the threat. Against a visual hunter like a fish, the decoy blob is most common. Against a chemical tracker like a moray eel, the diffuse cloud is more effective. The octopus adjusts its approach to match the predator, which once again requires threat assessment in real time. Octopuses also use ink offensively. Some species release a puff near prey to disorient it before striking. The ink disrupts the prey's senses and gives the octopus a crucial advantage during the attack. The ink sac is an ancient structure found across cephalopods. Fossil ink sacs have been found dating back to the Jurassic period, and in some cases, the preserved ink is still chemically active. Researchers have extracted melanin from these fossils and confirmed its structure is nearly identical to what modern octopuses produce. The recipe has barely changed in over 150 million years. What makes octopus ink truly impressive is how it fits into a layered defense system. Ink is never the first response. An octopus tries camouflage first. If that fails, it uses a startle display. If that fails, it flees by jet propulsion, squirting water through its siphon to rocket backward. Ink is deployed during the escape. If the predator still pushes through, the octopus may sacrifice an arm, leaving it wriggling on the seafloor as a final distraction. The arm grows back. Camouflage, startle, flee with ink and decoys, then arm sacrifice. Each layer requires different neural processing, different physical actions, and different chemical tools. The octopus switches between them seamlessly, in seconds, under life or death pressure. But all of these abilities raise a profound question. How did an animal this advanced end up sharing a family tree with snails and clams? The answer lies deep inside its genes, and what scientists found there was unlike anything they had ever seen in any other animal. There is a fossil sitting in a museum drawer that changes the way you think about everything we have discussed so far. It belongs to a creature called Pulsipia, and it is roughly 296 million years old. It has 8 arms, 2 eyes, what appears to be an ink sac. It is an octopus, or something very close to one, and it existed over 60 million years before the first dinosaur ever walked the earth. That number is difficult to absorb, so let us put it in perspective. Dinosaurs ruled the planet for about 165 million years. That feels like a staggeringly long time. Octopuses have been in the ocean for nearly twice that long. When the first Tyrannosaurus opened its jaws, octopuses had already been hunting, hiding, and evolving for over 200 million years. When the asteroid hit 66 million years ago and wiped out the dinosaurs, octopuses barely noticed. They were already ancient. They had survived mass extinctions before. They would survive this one too. The fossil record for octopuses is thin because their bodies contain almost no hard parts. Bones, teeth, and shells preserve well in rock. Soft tissue does not. To leave a fossil behind, an octopus had to die in exactly the right conditions, usually in very fine sediment that could capture the impression of its body before it decayed. These events were extraordinarily rare, which means the actual history of octopuses in the ocean is almost certainly far longer than the fossil record shows. Some scientists estimate that cephalopods as a group have been evolving for over 500 million years, stretching back to the Cambrian period, when complex animal life on Earth was just getting started. The octopus is not a newcomer, it is one of the oldest success stories in the history of life. The earliest cephalopods were not soft-bodied like modern octopuses. They had shells, large, coiled external shells that provided protection and buoyancy. The nautilus, which still exists today, is a living example of what those ancient cephalopods looked like. Over hundreds of millions of years, some cephalopod lineages began to lose their shells. The shell became internal, then smaller, then vanished altogether. Octopuses are the end point of that process. They traded armor for flexibility, protection for speed, rigidity for the ability to squeeze through any gap and become any shape. That trade-off was a gamble. Without a shell, octopuses became completely exposed to predators. Every shark, eel, dolphin, and large fish in the ocean could eat them. A shelled nautilus can retreat into its armor and wait out an attack. An octopus has no armor, nothing between its soft body and the teeth of a moray eel. In response, they evolved the most advanced camouflage system in the animal kingdom. The most sophisticated ink defense, the ability to squeeze into impossibly small spaces, and most importantly, the intelligence to outwit their enemies. Their brains got bigger. Their arms got smarter. Their survival strategy shifted entirely from armor to ingenuity. This is what scientists call an arms race between predator and prey, and octopuses ran it faster and further than almost any other invertebrate. Each generation that survived passed on slightly better camouflage, slightly quicker reaction times, slightly more flexible problem solving. Over 300 million years, those incremental improvements compounded into the extraordinary animal we see today. The octopus is what happens when evolution pressures a vulnerable animal to think its way out of danger for 300 million years straight. But the truly astounding discovery was not in the fossil record. It was in the living genome. When scientists finally cracked open the octopus genetic code and read it for the first time, they found something inside that nobody had predicted. A genome so strange, so scrambled, and so unexpectedly vast that it forced biologists to rewrite their understanding of how intelligence can be built. In 2015, an international team of scientists published the first complete genome sequence of an octopus. The species was the California two-spot octopus, and the results sent shockwaves through the genetics community. The octopus genome contains roughly 2.7 billion base pairs. That is nearly as large as the human genome, and it contains more than 33,000 protein-coding genes. That is more genes than a human has. Finding that many genes in a mollusk was unexpected. Clams, snails, and other relatives have much smaller, simpler genomes. Scientists initially suspected that the octopus genome had undergone whole genome duplication, a process where the entire set of genetic instructions accidentally copies itself. This is how some plants and fish acquired larger genomes, but when the researchers looked closely, they found that whole genome duplication had not occurred. The octopus genome grew large through a completely different mechanism. Instead of copying everything at once, the octopus genome expanded through the multiplication of specific gene families. One family in particular stood out: protocoderins. These are genes that regulate how neurons develop and how they form short-range connections with each other. In most invertebrates, there are a handful of protocoderin genes. Mammals have a moderate number. The octopus has 168. That is more than 10 times the number found in other invertebrates, and more than twice the number found in any mammal ever studied. Protocoderins are essential for building a complex nervous system. They control how nerve cells recognize each other, how they form connections, and how those connections are organized into functional circuits. Having 168 of them gave the octopus an enormous toolkit for wiring its brain and its arm nervous system in ways that no other invertebrate could match. Scientists believe this expansion was a critical step in the evolution of octopus intelligence. But the protocoderans were just the beginning of the surprises. The octopus genome is also riddled with an extraordinary number of zinc finger transcription factors, a family of genes involved in controlling when and where other genes are turned on and off. The octopus has roughly 1,800 of them. That is the second largest collection of this gene family ever found in any animal. The largest belongs to elephants, which have about 2,000. Most of the octopus zinc finger genes are active in embryonic and nervous tissue, suggesting they play a role in brain development. And then, there is the overall structure of the genome itself. In nearly every animal studied, related genes tend to cluster together on the same chromosome. Groups of genes that work together during development stay near each other, like co-workers in the same office. If you look at a fish genome, a mouse genome, or a human genome, you will find the same gene neighborhoods repeated over and over. Certain genes always live next to certain other genes. It is one of the most consistent patterns in genetics. The octopus genome has blown that arrangement apart. Genes that cluster together in virtually every other animal are scattered randomly across the octopus chromosomes. The HOX genes, a set of master genes that control body plan development in almost all animals, are normally found in a tidy row on one chromosome. In the octopus, they are spread across the genome with no apparent order. It is as if someone took a carefully organized library and threw every book onto a different shelf at random. Something shuffled the octopus genome like a deck of cards, and scientists believe they know what did it. Transposons, jumping genes, pieces of DNA that can cut themselves out of one location and paste themselves into another. The octopus genome is packed with them, with far more active transposons than most animals. And what these jumping genes do inside the octopus brain is one of the most startling genetic discoveries of the last decade. Inside your brain right now, buried in your DNA, are sequences of genetic code that can move. They can copy themselves and insert those copies into new locations across the genome. Scientists call them transposons, or jumping genes. They make up about 45% of the human genome. For a long time, most of them were considered genetic junk, leftover debris from millions of years of evolutionary accidents. Then, researchers started finding evidence that some of these jumping genes are not junk at all. They are active. And in the brain, they may play a role in learning and memory. One family of jumping genes in particular, called long interspersed nuclear elements, has drawn intense scientific attention. These sequences are still active in the human brain. They can create new copies of themselves and insert those copies into other genes, potentially changing how those genes function. Some researchers believe this process contributes to the diversity of neurons in the brain, giving each nerve cell a slightly different genetic profile. That neural diversity may be one of the foundations of human intelligence and cognitive flexibility. In 2022, a team of Italian researchers made a remarkable discovery. They found that the octopus brain is loaded with the exact same family of jumping genes. Not just the dormant, inactive kind, the active kind, the kind that are still jumping. The researchers analyzed two octopus species, the common octopus and the California two-spot octopus, and found active, long interspersed nuclear elements in the brain tissue of both. The jumping genes were most active in the vertical lobe of the octopus brain, a region associated with learning and memory. This was a stunning finding. Octopuses and humans diverged from a common ancestor over 500 million years ago. That ancestor was a simple, worm-like creature with no brain to speak of. The fact that both lineages independently evolved active jumping genes in their brain tissue, and that those genes belong to the same family, suggests a powerful case of convergent evolution at the genetic level. Two completely separate branches of the tree of life arrived at the same molecular solution for building complex brains. The researchers published their findings in the journal BMC Biology, and the paper quickly became one of the most discussed genetic studies of the year. The implication was clear. Jumping genes may not be random noise in the genome. They may be a fundamental ingredient of intelligence itself, appearing wherever evolution builds a sufficiently complex brain. The octopus is the strongest evidence yet that this genetic mechanism transcends the vertebrate world. There is a crucial detail to keep in mind. Most jumping genes in both humans and octopuses are dormant. They have been shut down by mutations or blocked by cellular defense mechanisms. Only a small fraction remain active, but that small fraction may be doing something enormously important. By inserting themselves into new locations, they create genetic variation between individual neurons. Your brain cells are not genetically identical to each other, and neither are an octopus's. This variation may be what gives both of our brains the flexibility to learn, adapt, and solve problems that we have never encountered before. No one predicted this connection. 500 million years of separate evolution, and both lineages independently activated the same family of jumping genes in their brain tissue. It is as if intelligence has a molecular signature, a genetic fingerprint that appears whenever evolution builds a mind complex enough to learn and remember. The octopus did not inherit this from us. We did not inherit it from them. Both lineages stumbled upon the same solution independently, which suggests it may be one of the few available routes to building a flexible, learning-capable brain. And yet, jumping genes are just one piece of the octopus genetic puzzle. Because octopuses have another ability at the molecular level that is far more dramatic, far more unusual, and far more important for their survival. They can rewrite their own genetic instructions in real time, and they do it on a scale that makes every other animal on Earth look frozen in place. Every cell in your body follows a simple chain of command. DNA holds the master blueprint. That blueprint gets copied into a temporary molecule called messenger RNA. The messenger RNA travels to the cell's protein-building machinery, which reads the instructions and assembles a protein. DNA to RNA to protein. That chain has been the central rule of molecular biology for decades. In almost every animal on Earth, the RNA is a faithful copy of the DNA. What the gene says, the RNA repeats, and the protein reflects. Octopuses break that rule. Inside the cells of an octopus, something happens to the messenger RNA after it leaves the nucleus. Enzymes intercept the RNA and change individual letters of the genetic code. They swap one chemical base, adenosine, for a different one, inosine, which the cell's machinery reads as guanosine. One letter changed. That single swap can alter the amino acid that gets inserted into the protein, which can change the shape of the protein, which can change its function entirely. This process is called RNA editing, and it is not unique to octopuses. Humans do it too. But in humans, RNA editing affects protein production in fewer than 3% of genes, and most of those edits occur in regions of RNA that do not code for proteins. They are minor tweaks with limited impact. In octopuses, RNA editing happens on a massive scale. More than 60% of the messenger RNA molecules in the octopus nervous system show evidence of editing. Tens of thousands of individual editing sites have been identified, and the majority of those edits occur in genes that code for proteins essential to brain function. This was first reported in 2015 by researchers at the Marine Biological Laboratory in Woods Hole, Massachusetts, and Tel Aviv University. They analyzed the RNA of the longfin inshore squid and found extensive editing. They then expanded their study to include octopuses and cuttlefish and found the same pattern across all the smart cephalopods. The nautilus, a shelled cephalopod with a much simpler brain, showed far less editing. The conclusion was striking. RNA editing scales with brain complexity in cephalopods. The smarter the animal, the more it edits. To put the scale in perspective, soft-bodied cephalopods edit RNA at tens of thousands of sites in their nervous systems. Humans edit at roughly 1,000 sites or fewer. Mice, fruit flies, and most other animals show similar low numbers. The octopus is operating on a completely different level. It is as if most animals are making a few pencil marks in the margins of their genetic textbook, while the octopus is rewriting entire chapters. What makes this even more remarkable is the trade-off involved. For RNA editing to work, the regions of DNA surrounding the editing sites must remain highly conserved. They cannot mutate freely because mutations would disrupt the editing machinery. This means that octopuses have sacrificed some of their capacity for DNA-level evolution in exchange for the ability to edit at the RNA level. They evolve more slowly in their genes, but adapt more quickly in their proteins. It is a completely different strategy for responding to the world, and it may be one of the reasons they are so successful. Think of it like this. Most animals adapt to their environment by slowly changing their DNA over many generations. That process takes hundreds or thousands of years. Octopuses found a shortcut. By editing RNA, they can produce different versions of the same protein on demand, within days, without changing a single letter of their DNA. The blueprint stays the same. The instructions change. The result is an animal that can reshape its own biology faster than any other complex organism on the planet. And the most dramatic demonstration of this ability comes from a single experiment that revealed just how fast and how far octopus RNA editing can go. In 2023, researchers published a study in the journal Cell that revealed the true scope of octopus RNA editing. The experiment was elegant and simple. They took California two-spot octopuses and placed half of them in warm water, around 72 degrees Fahrenheit, and the other half in cold water, around 55 degrees. They waited several weeks for the animals to adjust. Then they compared the RNA in the nervous systems of both groups. The results were staggering. The octopuses in cold water had edited their RNA at more than 13,000 individual sites, compared to the warm water group. 13,000 changes, almost all of them in the same direction, increasing the level of editing in cold conditions, and almost all of them targeted proteins involved in nervous system function. The octopuses were rewriting the operating instructions for their own brains in response to a temperature change. To understand why this matters, remember that octopuses cannot regulate their own body temperature. They are cold-blooded. When the ocean gets colder, every chemical reaction inside the octopus slows down. Enzymes work more slowly. Nerve signals travel more sluggishly. For an animal that depends on lightning-fast reflexes and split-second decision-making for survival, that slowdown could be fatal. The RNA editing appears to compensate for this. By changing the structure of key neural proteins, the octopus keeps its nervous system running at optimal speed, regardless of the water temperature. The researchers confirmed this by looking at two specific proteins in detail. The first was kinesin, a motor protein that transports cargo along the long branches of neurons. RNA editing changed the speed at which kinesin travels. The second was synaptotagmin, a protein that helps neurons communicate with each other at synapses. Editing altered the responsiveness of this protein. In both cases, the edited version of the protein performed better at cold temperatures than the unedited version. The team then wanted to know how quickly these changes happen. They took tiny juvenile octopuses and gradually changed the water temperature over 20 hours, adjusting it by about half a degree per hour. They tested the RNA before, immediately after, and 4 days later. The editing responded rapidly. Within hours, the octopuses had already begun adjusting their neural proteins. Within days, the changes were substantial. This is not evolution. This is not adaptation over generations. This is an individual animal remodeling its own brain chemistry in real time. To verify that this was not just a laboratory effect, the researchers caught wild octopuses during summer and winter along the California coast. The wild animals showed the same pattern. Winter octopuses had more RNA editing than summer octopuses. The results from nature matched the results from the lab. One of the lead researchers, Joshua Rosenthal, described it plainly. We are used to thinking that living things are programmed from birth with a fixed set of instructions. The idea that the environment can influence that genetic information, as we have shown in cephalopods, is a fundamentally new concept. Another researcher on the team, Eli Eisenberg, noted that no single example of this kind of environmentally responsive protein adjustment has ever been found in any mammal. Octopuses are doing something that our biology simply does not do. Compare this to how humans handle temperature changes. We shiver, we sweat, we put on a jacket. Our proteins stay the same, our RNA stays the same. The octopus responds to temperature change by rewriting the molecular instructions inside its nerve cells. It creates new protein versions that work better in the new conditions. It is not just adapting behavior, it is adapting biology. And it does this without any permanent change to its genetic code. The speed of this response is what makes it so extraordinary. DNA mutations take generations. Even gene expression changes through traditional mechanisms can take days or weeks to produce significant effects. RNA editing in octopuses happens within hours. The animal can retune its nervous system almost as fast as the environment changes around it. And the implications of this discovery extend far beyond marine biology. Because if RNA editing is the mechanism that allows octopuses to adapt their brains so quickly, it may also be the key to understanding how they became so intelligent in the first place. Here is the theory that is gaining traction among cephalopod researchers. RNA editing did not just help octopuses survive temperature changes. It may have been the driving force behind the evolution of their intelligence. Think about what RNA editing gives an animal. Instead of being locked into a single version of each protein, the octopus can produce multiple versions on demand. One gene can generate dozens of different protein variants depending on which editing sites are active. Multiply that across tens of thousands of editing sites, and the number of possible protein combinations becomes astronomical. The octopus nervous system is not running on a fixed set of instructions. It is running on a flexible, customizable library of proteins that can be reshuffled in response to any challenge. This flexibility could explain how octopuses achieve such complex behavior with relatively small central brains. Their neurons are not static. They are dynamic, constantly adjusting their own molecular machinery. A neuron involved in camouflage might express one set of protein variants, while a neuron involved in arm coordination expresses a different set from the same underlying genes. RNA editing provides a mechanism for this kind of targeted specialization without needing more genes or a bigger brain. Compare this to how vertebrates achieve protein diversity. In mammals, when evolution needs a new version of a protein, it typically duplicates the gene. Over time, the two copies diverge and take on different functions. This is effective but slow. It takes many generations and depends on random mutations. Octopuses rarely duplicate their genes. Their genome has far fewer gene duplications than you would expect for an animal with their level of complexity. Instead, they get their protein diversity from RNA editing. Same gene, many outputs. There is an evolutionary cost to this strategy. To preserve the RNA editing sites, the DNA around those sites must remain stable. That means the octopus genome evolves more slowly at the DNA level than you would predict. Researchers have confirmed this. Cephalopods that use extensive RNA editing show lower rates of DNA mutation in the regions surrounding editing sites. They have traded genetic evolution for something faster and more flexible. a kind of molecular agility that lets them adapt within a single lifetime rather than over generations. Some scientists have gone further and suggested that RNA editing may even contribute to individual variation in octopus behavior. If different octopuses edit their RNA differently in response to different life experiences, that could produce the distinct personalities that researchers have documented in captivity. One octopus might be bold because its neural proteins are tuned one way. Another might be shy because its proteins are tuned differently. Same genes, different edits, different personalities. This is speculative, and it should be labeled as such. The connection between RNA editing and personality has not been directly demonstrated, but the theoretical framework is sound, and several research groups are actively investigating it. If confirmed, it would mean that octopuses are not just products of their genes and their environment. They are, in a very real molecular sense, self-editing organisms. They participate in their own biological customization. There are also practical implications for human medicine. Researchers are already exploring whether the RNA editing tools used by octopuses could be adapted for therapeutic use in humans. If scientists could harness the same enzymatic machinery that octopuses use to swap out single letters of RNA, they might be able to correct harmful mutations without permanently altering DNA. The octopus approach is temporary, reversible, and targeted. Those are exactly the qualities that medical researchers want in a gene therapy tool. Joshua Rosenthal and his colleagues have received funding from the National Institutes of Health to investigate whether cephalopod RNA editing could lead to non-addictive treatments for pain and other conditions. And all of this happened in a lineage that split away from ours over 500 million years ago. The intelligence we see in an octopus was not inherited from a shared smart ancestor. It was built from scratch on a completely independent branch of the evolutionary tree. That fact alone carries implications that reach far beyond the ocean floor. The last common ancestor of humans and octopuses lived roughly 550 million years ago during a period called the Ediacaran, just before the Cambrian explosion that gave rise to most of the major animal groups alive today. That ancestor was almost certainly a simple, worm-like creature. It had no eyes, no brain, no arms, no complex behavior. It was a flat, soft-bodied organism drifting or crawling along an ancient seafloor, and it gave rise to two enormously different lineages. One lineage led to the vertebrates: fish, amphibians, reptiles, birds, and mammals. That line organized its nervous system into a spinal cord running up the back, topped by an increasingly large and centralized brain. Over hundreds of millions of years, this design produced dolphins, crows, primates, and eventually humans. The brain sat at the top, the body followed orders, and intelligence was concentrated in one organ. The other lineage led to the mollusks: clams, snails, slugs, and eventually cephalopods. This line took a radically different path. Instead of centralizing the nervous system, cephalopods distributed it across the body. Instead of building a spine, they stayed boneless. Instead of growing a skull to protect a single brain, they spread their neurons across their entire body. And from that fundamentally different architecture, working with completely different building blocks, they built their own version of intelligence. This is what makes octopus intelligence so scientifically valuable. It is not a variation on our intelligence. It is an entirely independent invention. When a crow uses a tool, we can trace that ability back through a shared evolutionary heritage with mammals. Both crows and humans descended from animals that already had complex brains. The wiring is different, but the foundation is similar. Octopuses share no such foundation with us. Their intelligence evolved from scratch, using different genes, different brain structures, and different body plans. Scientists call this convergent evolution. It is the process by which unrelated organisms independently arrive at similar solutions to similar problems. Wings evolved separately in birds, bats, and insects. Eyes evolved separately in vertebrates and mollusks. Echolocation evolved separately in bats and dolphins. And intelligence, it now appears, evolved separately in vertebrates and cephalopods. The fact that evolution built complex cognition twice, using completely different raw materials, suggests that intelligence may be a likely outcome whenever nervous systems reach a certain level of complexity. It may not be a lucky accident. It may be an almost inevitable product of enough neurons under enough pressure. That idea has profound implications for the search for life beyond Earth. If intelligence is not a one-time accident, but a repeatable product of evolution under the right conditions, then the odds of finding intelligent life on other planets go up dramatically. The octopus is the strongest evidence we have that smart minds can emerge from bodies nothing like our own, using genes nothing like our own, following evolutionary paths nothing like our own. If it happened independently on this planet, in the ocean, in a mollusk, there is no reason it could not happen on another world in an entirely different form. One prominent researcher put it this way: studying the octopus is the closest we can come to meeting an intelligent alien without leaving our planet. That quote has been repeated so often in cephalopod science that it has become almost a cliche, but it carries real scientific weight because the octopus represents a genuine alternative model for how intelligence can be organized. It is not a lesser version of our intelligence. It is a different version, built from different parts, running on different fuel, solving the same fundamental problems of survival in ways we are only beginning to understand. And nowhere is that alternative design more visible than in one specific organ. An organ that looks almost identical to a human eye, but was built from a completely different set of evolutionary instructions. An octopus eye looks remarkably like a human eye. It has a lens. It has a retina. It has an iris that can expand and contract to control how much light enters. If you put a diagram of an octopus eye next to a diagram of a human eye, the similarity is striking. And yet these two eyes share no common ancestor that had anything resembling an eye. They were designed independently by evolution, separated by over 500 million years of divergent history. This is one of the most famous examples of convergent evolution in biology. Both octopuses and humans needed to solve the same problem: how to form sharp images in an environment filled with useful visual information, and both arrived at nearly the same solution: a camera-style eye with a single lens that focuses light onto a sheet of photoreceptor cells. But the octopus eye has one key difference that makes it, in some ways, superior to the human version. It has no blind spot. In the human eye, the optic nerve, the cable that carries visual information from the retina to the brain, connects to the front side of the retina. This means the nerve fibers have to pass through the retina and gather into a bundle that exits through a hole in the back. That hole creates a small area where there are no photoreceptors. That is the blind spot. Your brain fills it in so seamlessly that you never notice it. But it is there. It is a design flaw that exists because of how vertebrate eyes evolved. The wiring went in from the front, and the nerve exits from the back. The octopus eye evolved its retina the other way around. The photoreceptors face the incoming light, and the nerve fibers connect behind them. The optic nerve exits from the back of the retina without ever passing through it. No hole, no blind spot. If an engineer were designing an eye from scratch, this is how they would do it. The octopus got it right. Vertebrates got it slightly wrong. This structural difference is powerful evidence that octopus eyes were not inherited from a common ancestor with vertebrates. If they were, the wiring would be the same. Instead, evolution ran the same experiment twice with slightly different construction methods, and the octopus version avoided the one flaw that the vertebrate version could not. Despite this superior structural design, octopus eyes have their own limitations. As we discussed in earlier chapters, they have only one type of photoreceptor, which should make them colorblind. Vertebrate eyes evolved multiple photoreceptor types to see color. The octopus eye took a different path, possibly compensating through chromatic aberration, skin-based light sensing, or other mechanisms that remain under investigation. Octopus eyes also process visual information differently than ours. In vertebrates, the retina does some initial processing before sending data to the brain. In octopuses, the optic lobes behind the eyes are enormous, taking up a significant portion of the central brain. These lobes handle a huge amount of visual processing, which may explain how octopuses can detect and respond to visual patterns so quickly, despite their relatively simple photoreceptors. The convergent evolution of octopus and human eyes tells us something important about the nature of biological solutions. When evolution faces the same challenge repeatedly, it often converges on similar designs. Lenses, retinas, and camera-style optics appear to be an almost inevitable outcome when an organism needs high-resolution vision in a light-rich environment. The specific wiring may differ, but the architecture is remarkably conserved across lineages that have been separated for longer than most life has existed on this planet. And that is what makes the octopus so valuable to science. It is a parallel experiment, a second attempt, a different answer to the same question. Everything that is similar between us and them tells us about the fundamental rules of biology. Everything that is different tells us about the range of possibilities. But there is one thing about the octopus that seems like a cruel evolutionary mistake. An animal this intelligent, this capable, this remarkable in its biology, lives an astonishingly short life. And what happens during that brief existence may be the most haunting part of their story. In 2021, a team of Brazilian scientists published a study that added another layer to the growing mystery of octopus consciousness. They filmed sleeping octopuses in their laboratory and observed something mesmerizing. During certain phases of sleep, the octopuses erupted with rapid color changes across their skin. Bursts of orange, flashes of brown, ripples of pale white. Their eyes twitched. Their suckers contracted. Their arms moved in small, jerky motions. Then, just as quickly as it started, the activity stopped. The octopus settled back into stillness and went pale again. The scientists identified two distinct stages of sleep. During what they called quiet sleep, the octopus lay still with pale skin and tightly contracted pupils, completely unresponsive to mild stimulation. During active sleep, the skin display fired up, the body twitched, and the octopus became briefly animated while remaining genuinely asleep. The researchers tested this by presenting sleeping octopuses with a video of a swimming crab, one of their favorite foods. During both sleep stages, the animals did not respond unless the stimulus was very strong. They were truly asleep. This pattern has a striking parallel. In humans, sleep alternates between quiet non-REM stages and active REM stages. REM is the phase where most dreaming occurs. During REM, our eyes move rapidly, our muscles twitch, and our brain activity looks almost identical to when we are awake. The octopus active sleep phase shares several of these features: eye movement, muscle twitching, brain-like skin activity, and it occurs after a long period of quiet sleep, just as REM follows deep, non-REM sleep in mammals. A 2023 study published in Nature took this even further. Researchers at the Okinawa Institute of Science and Technology recorded brain activity in sleeping octopuses and found that during active sleep, the neural patterns closely resembled those seen when the octopus was awake. They also found brainwave patterns during quiet sleep that resembled mammalian sleep spindles, waveforms believed to be involved in memory consolidation. These spindle-like waves occurred in brain regions associated with learning and memory. The researchers also tested whether active sleep is biologically necessary by gently disturbing sleeping octopuses every 2 to 3 minutes for 48 hours. Sleep-deprived octopuses entered active sleep more frequently and more quickly once they were allowed to rest. This compensatory behavior is called sleep rebound, and it is a hallmark of essential sleep stages in mammals. The octopus needs its active sleep. It is not optional. The most tantalizing observation came from high-resolution filming of the skin patterns during active sleep. The color changes were not random. They cycled through recognizable patterns, the same camouflage sequences the octopus used while awake. Hunting patterns, hiding patterns, threat displays. It was as if the sleeping octopus was replaying its waking experiences on its skin. One researcher described the skin as a visual readout of brain activity during sleep. Are octopuses dreaming? Scientists cannot say for certain. Dreams require subjective experience, and we cannot ask an octopus what it perceives. But the evidence is building that during active sleep, octopuses are processing and potentially rehearsing their waking experiences. If that constitutes dreaming, then octopuses dream in color, on their skin, for all the world to see. This discovery carries enormous weight for our understanding of sleep and consciousness. It means that two-stage sleep, with quiet and active phases, evolved independently in vertebrates and in cephalopods, separated by over 500 million years of evolutionary history. Whatever function this type of sleep serves, it appears to be so important that evolution invented it twice. An animal that may dream, an animal that edits its own genes, an animal with 9 brains and blue blood, and it all ends in 1 to 2 years. The question of why burns at the heart of octopus? Science. And the answer involves a tiny gland and a death sentence written into its own biology. Here is the cruelest fact in all of marine biology. An animal with the intelligence to solve puzzles, use tools, recognize human faces, and possibly dream lives an average of 1 to 2 years. Some species survive a little longer. The giant Pacific octopus, the largest species in the world, can live up to 5 years under ideal conditions. But most octopuses hatch, grow, learn everything from scratch with no parental guidance, reproduce once, and die. All within the span of 1 to 2 trips around the sun. Compare that to other intelligent animals. Dolphins live 40 to 60 years, elephants live 60 to 70, crows live 10 to 15, even parrots can live for decades. These animals have long lifespans partly because they live in social groups, where knowledge is passed between generations. A young dolphin learns from its mother. A young crow learns from its flock. Intelligence in these species is cumulative. It builds over years of experience and social learning. Octopuses get none of that. Their mothers die before they hatch. Their fathers are long gone. No octopus has ever been taught anything by another octopus. Every skill, every hunting technique, every camouflage pattern, every problem-solving trick that an octopus knows was learned entirely on its own, from zero, in a matter of months. That is one of the most remarkable achievements of individual learning in the animal kingdom. Imagine if you were born alone, with no parents, no teachers, no books, and no one to ask for help, and you had to figure out everything about the world by yourself before your first birthday. That is the life of every octopus. The speed of that learning is extraordinary. Juvenile octopuses emerge from their eggs already capable of changing color and squirting ink. Within weeks, they are hunting live prey. Within months, they are solving problems that take laboratory researchers hours to design. They learn at a pace that suggests their brains are wired for rapid acquisition of information, which may be related to the RNA editing we discussed earlier. Their neurons can be customized in real time, potentially accelerating the learning process at the molecular level. Every day of an octopus's life is a crash course in survival, and they pass with flying colors. But why the short lifespan? Why would evolution build such an extraordinary mind and then limit it to such a brief window? The answer appears to involve a reproductive strategy that prioritizes the next generation at the expense of the current one. Most octopus species are what biologists call semiparous. They reproduce once and then die. This is not an accident or a disease. It is a programmed biological event. After mating, a cascade of hormonal changes sweeps through the octopus body. shutting down essential functions one by one. Digestion stops, the immune system collapses, muscles deteriorate, behavior becomes erratic. The animal enters a terminal decline that no amount of food or care can reverse. This strategy is rare among intelligent animals, but common in certain fish and insects. Salmon are the most famous example. They swim upstream, spawn, and die within days. The theory behind semelparity is that by investing all available energy into a single, massive reproductive event, the animal maximizes the number of offspring that survive. There is no energy wasted on maintaining an aging body. Everything goes toward eggs and their protection. It is brutally efficient. For octopuses, this means a short but intense life. Learn fast, hunt well, reproduce once, die. It is a strategy that works brilliantly from an evolutionary standpoint, but it creates a heartbreaking paradox. The most intelligent invertebrate on Earth never gets to accumulate wisdom over a long life. Every octopus genius dies young. Every lesson learned is lost forever. And the organ responsible for triggering, the death is so small, you could miss it with the naked eye. Between the eyes of every octopus sits a tiny organ called the optic gland. It is roughly the size of a grain of rice. It sits near the optic nerve and functions similarly to the pituitary gland in mammals, acting as a chemical messenger between the brain and the rest of the body. For most of an octopus's life, this gland operates quietly in the background, helping regulate growth and development. But after mating, something changes inside it. The optic gland activates what scientists have grimly nicknamed the self-destruct system. In 1977, a researcher named Jerome Wodinsky at Brandeis University performed an experiment that proved the optic gland's deadly role. He surgically removed the optic glands from female Caribbean two-spot octopuses that had just laid eggs and were already refusing food. The result was immediate and dramatic. The females abandoned their eggs. They started eating again. They gained weight. Some of them mated a second time. They lived for months longer than they would have otherwise. Removing the gland reversed the death sentence. This confirmed that the optic gland was not just associated with reproductive behavior. It was actively causing the death spiral. Something the gland produced after mating was shutting down the digestive system, destroying the immune system, and driving the octopus towards self-destruction. For decades, the exact mechanism remained unclear. Scientists knew the gland was responsible but could not identify the specific molecules involved. The gland was too small, too difficult to isolate, and the technology to analyze its chemical output did not exist yet. Then, in 2018, researcher Zi-Yan Wang at the University of Chicago sequenced the RNA of optic glands from female octopuses at different stages of their post-mating decline. She found higher levels of activity in genes related to sex hormones, insulin-like hormones, and cholesterol metabolism. As the animals declined, their optic glands were flooding their bodies with a complex cocktail of hormones that disrupted normal function at every level. In 2022, Wang and her colleagues went further. They directly analyzed the chemicals produced by the optic gland and identified 3 distinct pathways, all related to cholesterol. One pathway produced pregnenolone and progesterone, steroids commonly associated with pregnancy. A second produced precursors for bile acids. A third produced high levels of a cholesterol precursor called 7-dehydrocholesterol, which is known to be toxic at elevated concentrations in humans. In fact, in a rare genetic disorder called Smith-Lemli-Opitz syndrome, high levels of this same molecule cause severe developmental problems and cognitive impairment in human children. The octopus optic gland was flooding the animal's body with a molecule that is essentially a poison. The gland was not secreting a single death hormone. It was triggering multiple biochemical cascades simultaneously, each one contributing to a different aspect of the decline. This multiplicity may explain why the death spiral involves so many different symptoms. The octopus does not just stop eating, it loses coordination. Its skin dulls and loses its ability to change color. its wounds stop healing. Its behavior becomes erratic and self-destructive. In captivity, some females in the late stages of decline have been observed banging against tank walls, tearing off pieces of their own skin, and eating the tips of their own arms. These are not survival behaviors. They are symptoms of a system-wide biochemical collapse, triggered by a gland the size of a grain of rice. The male octopus faces a similar fate after mating, though the mechanism is less well studied. Males stop eating, become disoriented, lose their camouflage ability, and often fall prey to predators within weeks of mating. Whether the same optic gland pathways drive the male decline remains an open question. What is clear is that the optic gland sits at the center of one of nature's most dramatic trade-offs. An organ that ensures reproductive success also guarantees death. And for the female octopus, the weeks between mating and dying contain one of the most extraordinary acts of devotion in the entire animal kingdom. A female octopus does not just lay her eggs and walk away, she guards them. For weeks. For months. In some deep-sea species, for years. And during that entire vigil, she does not eat a single thing. The common octopus lays tens of thousands of eggs, each one tiny, translucent, and attached to the ceiling of her den in long braided strands. From the moment those eggs are laid, the mother begins her watch. She uses her arms and siphon to blow fresh, oxygenated water across them. She cleans them, removing algae and bacteria. She defends them from predators. She does this every hour of every day for the entire incubation period, which lasts 1 to 3 months, depending on species and water temperature. During this time, she refuses food. Researchers have placed live crabs within arm's reach of brooding females, and the mothers did not eat. They stayed on the eggs. Their bodies begin to consume themselves, breaking down muscle tissue for energy. They visibly shrink. Their skin loses color. Their movements become slower. By the time the eggs hatch, many mothers are barely alive. In the most extreme documented case, a deep-sea octopus was observed brooding her eggs for 4.5 years. Researchers, using a remotely operated vehicle in the Monterey Canyon, off the coast of California, spotted the female in April of 2007, clinging to a rock ledge at a depth of nearly 5,000 feet. They returned to the same spot over the next 53 months, 18 separate visits, and found her in the same position every time, guarding the same clutch of eggs. She never moved. She never ate. Over the years, her skin turned pale and scarred. When they returned for the final visit, she was gone and the rock was covered in empty egg casings. The eggs had hatched. The mother had died. 4.5 years without food. In cold, dark water nearly a mile below the waves. That is the longest brooding period ever recorded for any animal on Earth. Confirmed and published in the journal PLOS1. Why does she do this? The eggs are vulnerable. Without constant cleaning, they would be overgrown with bacteria and fungus. Without a stream of oxygenated water, the embryos inside would suffocate. Without a guardian, predators would eat them. The mother is the only thing standing between her offspring and oblivion. Her sacrifice is not symbolic. It is physically necessary for the eggs to survive. The tragedy is made worse by a specific detail of octopus development. Octopus mothers never meet their children. The eggs hatch after the mother dies, or in most cases the mother dies within days of the eggs hatching. The baby octopuses emerge into the ocean completely alone. They are the size of grains of rice. They drift in the water column, carried by currents, feeding on microscopic plankton. Most of them will be eaten within their first weeks of life. Of the tens of thousands of eggs a female lays, only a handful of offspring will survive to adulthood. The mother gives everything so that a few might live. The evolutionary logic behind this sacrifice is cold, but effective. By refusing to eat, the mother ensures she stays at the den. If she left to hunt, Predators would find and eat the eggs within minutes. The eggs have no defense on their own. They cannot swim away. They cannot hide. They cannot change color. Without a guardian, they are simply food for anything that finds them. The mother is the barrier between her offspring and extinction, and she pays for that barrier with her life. In some species, the mother assists the hatching process, gently blowing water over the eggs as they begin to break open. She watches as the first of her children swim away, and then, already dying, she lets go. Every octopus alive today exists because its mother starved herself to death, protecting eggs she would never see hatch. That fact alone should change how we think about these animals. But evolution, as it often does, left open one exception. One species that broke the rule. One octopus that mates, survives, and mates again. And scientists are racing to understand why. In the shallow waters of the Pacific coast, from Nicaragua down to Panama, lives a small octopus barely the size of a thumb. It has pale skin marked with bold dark stripes running the length of its body. Its name is the lesser Pacific striped octopus, and it is the most important octopus in the world right now for one simple reason. It does not die after mating. Every other well-studied octopus species follows the same tragic pattern. Mate, brood eggs, decline, die. The lesser Pacific striped. The octopus breaks that cycle. Females lay multiple clutches of eggs over their lifetime. They continue eating while brooding. They mate repeatedly with multiple partners. They do not self-destruct. They live significantly longer than their semiparous relatives, and they can reproduce more than once. This was first described by researchers who studied the species in the early 1970s, but the observations were largely ignored by the scientific community because they contradicted everything known about octopus biology. It took decades before the species was bred successfully in captivity, which finally happened in 2019 at the Marine Biological Laboratory in Woods Hole, Massachusetts. That achievement, the first successful multi-generational breeding of this species in a lab setting, opened the door to rigorous scientific study. The critical question is obvious. Why does this species survive while all others die? What is different about its optic gland? What is different about its hormonal pathways? If scientists can identify the specific molecular mechanism that prevents the self-destruct cascade in this octopus, it could reveal fundamental insights about aging, reproduction, and the trade-offs between them, not just in octopuses, but potentially in all animals. Researcher Zi Yan Wang, who mapped the death-triggering pathways in other octopus species, has specifically stated that comparing the optic glands of the striped octopus to those of single mating species is her next major research goal. The striped octopus has an optic gland just like every other cephalopod. The gland is there. It simply does not trigger the same cascade. Something in its biology diffuses the bomb. The lesser Pacific striped octopus is also unusual in its social behavior. Unlike most octopuses, which are fiercely solitary, these animals tolerate each other's company. Mating pairs have been observed sharing dens. Females do not attack males during mating, which is common in other species. They even engage in a unique mating behavior where the male and female press their bodies together beak to beak, a position that would be suicidal with most other octopus species, because the female would typically try to kill and eat the male. This combination of traits, multiple mating, extended lifespan, social tolerance, and non-lethal mating, makes the lesser Pacific striped octopus an almost perfect model organism for studying the evolution of lifespan and social behavior. It is small enough to breed easily in the lab. It reproduces frequently enough to generate research populations. And it is different enough from other octopuses to provide meaningful comparisons. Some scientists have speculated about what would happen if octopuses in general evolved longer lifespans and the ability to reproduce more than once. With their intelligence, their learning speed, and their problem-solving abilities, a longer-lived octopus could potentially accumulate knowledge over years instead of months. It might develop more complex behaviors, form stable relationships, and perhaps even pass information between generations. The lesser Pacific-striped octopus hints at what that alternative evolutionary trajectory might look like. It is a living glimpse of what octopuses could become if the self-destruct mechanism were removed. There is also a practical consideration. Because this species mates multiple times and tolerates social contact, it is far easier to maintain in a laboratory setting than solitary, single mating species. Researchers do not have to constantly collect new animals from the wild. They can breed colonies and study multiple generations. This makes the lesser Pacific-striped octopus an almost ideal model organism for understanding cephalopod biology at a deeper level. And that possibility became even more intriguing when scientists discovered that octopuses are not always the solitary loners we thought they were. In one remarkable location, they had already started building something that looked disturbingly like a neighborhood. In 2012, divers exploring Jervis Bay off the coast of New South Wales, Australia, discovered something that should not have existed. On a patch of sandy ocean floor, about 50 feet below the waves, as many as 16 octopuses were living together. Not temporarily, not by accident. They had built dens side by side in a mound of discarded shells, and they were interacting with each other constantly, mating, fighting, communicating through color changes, evicting each other from dens. Scientists named the site Octopuses. The initial reaction from the research community was skepticism. Octopuses are supposed to be solitary. They are supposed to avoid each other except during mating. They are supposed to be loners who tolerate no company. Octopuses contradicted all of that. Some researchers dismissed it as an anomaly caused by a piece of human-made metal debris at the center of the site, which may have attracted the first octopus and inadvertently created a gathering point. Then, in 2016, a second site was discovered nearby. Researchers led by David Scheel from Alaska Pacific University found another octopus settlement, just a few hundred yards away from Octopuses. This one had no human debris at its center. It was built entirely from natural materials, mainly scallop shells and rock outcrops. It contained 23 dens and hosted up to 15 gloomy octopuses at a time. They named it Octlantis. Over 8 days of observation using underwater cameras, the researchers recorded an astonishing range of social interactions. Octopuses fought over dens. They chased each other across the shell bed. They darkened their skin to signal aggression and paled to signal submission. They mated. They evicted rivals from their homes. They modified their environment by piling shells around their dens to reinforce them. The footage looked, in the words of one journalist, like an underwater soap opera. A 2022 study added another remarkable behavior. Researchers documented octopuses at these sites throwing debris at each other. They would gather silt, shells, or algae into their arms and propel it through the water by jetting with their siphon. The throws were directed at other octopuses, and they hit their targets about 17% of the time. Females were the most frequent throwers, often targeting males who had overstayed their welcome after mating attempts. Some throws appeared to be defensive, others seemed motivated by pure irritation. Intentional throwing of projectiles at other individuals is extremely rare in the animal kingdom, previously documented only in a handful of primate and elephant species. Finding it in a mollusk was another reminder that octopus behavior keeps breaking the rules we thought applied to all invertebrates. The researchers were careful to note that these settlements are not cities in any human sense. There is no cooperation, no shared infrastructure, no division of labor. The octopuses are not building a community together. They are individual animals competing for limited den space in a food-rich area. Their proximity is driven by ecology, not by social bonds. But the fact that they can tolerate each other communicate through visual signals, and engage in complex social interactions like eviction, signaling, and projectile throwing, is a significant departure from the old assumption that octopuses are incapable of social behavior. Scheel, the lead researcher, put it simply: these behaviors are the product of natural selection and may be remarkably similar to complex social behavior in vertebrates. When the right conditions occur, evolution may produce very similar outcomes in diverse groups of organisms. The discovery of Octopus and Octlantis suggests that the solitary octopus may be solitary by circumstance, not by nature. Given enough food and limited shelter, they will congregate, interact, and behave in ways that we previously reserved for animals with much larger brains and much longer lifespans. And it is precisely this kind of complexity, this undeniable intelligence, that has made the growing global push to farm octopuses one of the most fiercely debated ethical questions in marine science today. In recent years, several companies and governments around the world have announced plans to farm octopuses commercially. The demand for octopus meat has grown dramatically, particularly in Mediterranean, East Asian, and Latin American markets. Wild catch alone can no longer keep up. The logical business response is aquaculture. Breed them in captivity, grow them in tanks, harvest them at scale. But the scientific community has pushed back with a force that has made international headlines. The objections are not primarily environmental, though those concerns exist. The objections are moral. Can we ethically farm an animal that solves puzzles, uses tools, recognizes individual humans, exhibits play behavior, edits its own genes, and may even dream? In 2024, over 500 researchers signed a declaration supporting the likelihood of consciousness in mammals and birds, and acknowledging the possibility of consciousness in cephalopods. This followed the 2012 Cambridge Declaration on Consciousness, which stated that many non-human animals possess the neurological substrates that generate consciousness. Octopuses were specifically mentioned. The scientific consensus is moving, slowly but steadily, toward recognizing that octopuses experience something. Whether that something qualifies as suffering in the way humans understand it remains debated. But the direction of the evidence is clear. The practical problems with octopus farming are also severe. Octopuses are solitary and territorial. Keeping them in close quarters leads to stress, aggression, and cannibalism. They are escape artists that defeat containment systems. They require varied diets of live or freshly killed prey, which is expensive and difficult to provide at scale. They are prone to self-harm when stressed, biting their own arms or refusing food. And they have short lifespans, which means the turnover rate in a farming operation would be enormous. Several countries have already taken legislative action. The United Kingdom passed the Animal Welfare Sentience Act in 2022, which officially recognized octopuses and other cephalopods as sentient beings under law. This means any octopus farming operation in the UK would be subject to the same welfare standards applied to farming vertebrate animals. Similar legislation is being considered in other European countries. The state of Washington in the United States banned octopus farming outright. Other jurisdictions are considering similar measures. Meanwhile, a Spanish company announced plans to open the world's first commercial octopus farm, sparking protests from animal welfare organizations and condemnation from dozens of marine scientists. The debate cuts to the heart of a question that humans have been avoiding for centuries. Where do we draw the line on which animals are too intelligent to eat? We have drawn that line for dogs, for horses, for apes, and for dolphins in various cultures. Now, science is asking whether the octopus belongs on that protected list. The more we learn about their intelligence, their emotional range, and their capacity for suffering, the harder it becomes to treat them as mere seafood. Proponents of farming argue that sustainable aquaculture reduces pressure on wild populations and provides food security for communities that have eaten octopus for thousands of years. They point out that we farm pigs, which are also highly intelligent, and that intelligence alone should not determine which animals we eat. Critics argue that the intelligence, sentience, and individual personality of octopuses make them categorically different from fish or shellfish, and that the conditions required for farming would inevitably cause suffering. An animal that solves puzzles when bored, that hurls objects at individuals it dislikes, and that tears off its own skin when stressed, is not an animal that will thrive in a concrete tank. The debate is ongoing, and the science is moving faster than the policy. New studies on octopus cognition are published almost monthly, each one adding to the weight of evidence that these animals experience the world in ways that matter morally. The question is no longer whether octopuses are intelligent. The question is what we are willing to do about it. What is not debatable is what octopuses have taught us about the nature of mind itself. And that lesson, more than any other, is why this animal matters far beyond the ocean. In 2025, a study confirmed that octopuses experience something resembling the rubber hand illusion, a test of body awareness, and self-perception previously thought to be limited to humans and a small number of mammals. When a false arm was stroked alongside their real arm, the octopuses showed a response indicating they could be fooled into perceiving the fake as their own. They have a sense of self. They know what belongs to them. Add this to the list. They solve puzzles, they use tools, they play, they recognize faces, they hold grudges, they dream, they edit their own genes, they have 9 processing centers spread across their body, they have personalities, they build structures, they throw things at each other, and they experience something that scientists are increasingly willing to call awareness. The word that keeps appearing in the literature is consciousness. Not human consciousness. Not mammalian consciousness. Something else. Something built from different materials, running on different hardware, operating through a distributed network rather than a centralized processor. If consciousness in humans is like a desktop computer with one powerful chip, consciousness in an octopus may be like a network of smaller processors working in parallel, each one contributing a piece to the whole. The philosopher Peter Godfrey Smith, who has spent years studying octopus cognition, described the encounter this way. Our most recent common ancestor with the octopus is so distant, more than twice as ancient as the first dinosaurs, that if we can connect with them as sentient beings, it is not because of a shared history. It is because evolution built minds twice over. They are probably the closest we will come to meeting an intelligent alien. That framing has enormous implications. If intelligence and awareness can arise from a body plan so radically different from ours, then the range of possible minds in the universe is far wider than we imagined. We tend to picture alien intelligence as something roughly humanoid, a brain in a head on a body with limbs. The octopus suggests that intelligence might take shapes we would not recognize at first glance. Distributed, embodied, woven into skin and muscle, tasting the world with every touch. The octopus also challenges our understanding of what intelligence requires. We assumed it required social life. Octopuses are loners. We assumed it required a long lifespan. Octopuses live 1 to 2 years. We assumed it required parental teaching. Octopuses are orphans from birth. We assumed it required a centralized brain. Octopuses spread their cognition across 9 centers. We assumed it required a skeleton to support a large brain. Octopuses have no bones at all. Every assumption we made about the prerequisites for intelligence, the octopus has violated. One by one, it has knocked down the pillars we built our understanding on and replaced them with something stranger and more wonderful. And perhaps that is the most important thing they teach us. Intelligence is not one thing. It is not a single ladder with humans at the top. It is a landscape with many peaks, many pathways, and many possible forms. The octopus reached its peak through a route that no other animal has taken, using tools no other animal uses, in a body no other intelligent animal shares. It is proof that the universe has more than one way to build a thinking creature. We were wrong about them. For centuries, we grouped them with the simplest creatures in the ocean. We assumed a boneless animal could not think. We believed intelligence required a spine, a skull, and a social group. The octopus proved all of it wrong, and it did so quietly, alone, in a body that dissolves when it dies, leaving behind nothing but a beak and an empty den. The next time you see an octopus, whether in a documentary, an aquarium, or on a dive, remember what you were looking at. You are looking at 500 million years of independent evolution. You are looking at an animal that rewrites its own brain. You are looking at something as close to an alien intelligence as exists on this planet. And you are looking at a creature that, despite everything science has learned, still holds secrets we have not yet begun to unravel. Every octopus that has ever lived discovered the world on its own. No mother showed it how to hunt. No father taught it to hide. No older sibling demonstrated how to open a shell or change color or squeeze through a gap the width of a coin. Every single octopus, every one of the roughly 300 known species, started from zero, and in 12 to 24 months, each one built a working understanding of a world that would challenge any mind, human or otherwise. Then it mated, and it died, and everything it learned died with it. That is the tragedy at the center of this story. Intelligence without inheritance, genius without legacy. Every generation reinvents the wheel, masters its world, reproduces, and vanishes. No culture accumulates, no wisdom passes forward. The octopus is proof that a brilliant mind can exist in total isolation, and that evolution does not care whether that brilliance survives the individual. But what the octopus leaves behind for us is something different. It leaves a mirror. Not a reflection of what we are, but a reflection of what intelligence can be when it takes a completely different form. When it runs on copper blood instead of iron. When it thinks with its arms instead of its head. When it rewrites its own genetic instructions instead of waiting for evolution to do it. When it sees without color and matches color perfectly. When it dreams in patterns on its skin. The octopus is the strongest argument science has ever produced that mind is not an accident. It evolved independently, at least twice, on this one small planet, in bodies that could not be more different. If it happened here, in the cold dark water at the bottom of a reef, it can happen anywhere. Every year, researchers learn something new about these animals that forces them to redraw the boundaries of what is possible in biology. The RNA editing alone has implications for human medicine. Scientists are already exploring whether the same molecular tools that octopuses use to customize their brain proteins could be adapted to develop new therapies for genetic diseases and neurological conditions. The distributed intelligence of the octopus is inspiring new approaches to robotics and artificial intelligence. The camouflage system is being studied for applications in military technology and adaptive materials. But beyond the practical applications, the octopus offers something harder to measure and arguably more important. It offers humility. For centuries, we placed ourselves at the top of an intelligence hierarchy and arranged every other living thing below us in descending order of worth. The octopus does not fit on that ladder. It sits outside it entirely, on a branch of the tree of life so distant from ours that finding intelligence there is like finding a familiar word in a language you have never heard. We were wrong about the octopuses. We underestimated them for so long that we nearly missed one of the most important stories in the history of life on Earth. A story about a creature that taught itself everything, in a body made of nothing but nerve and muscle, in a life shorter than a single school year. And somewhere right now, on a reef you have never seen, in water you will never touch, a young octopus is hatching from an egg the size of a grain of rice. It is alone. Its mother is dead. It has never seen another octopus. It has no idea what it is or what it can do. But within months, it will be solving problems, making decisions, and navigating a world of predators and prey with a sophistication that took mammals millions of years to develop. It will live fast. It will learn everything. And then it will be gone. That is the octopus. That is what we were wrong about. And that is why they matter.